Hello there, and welcome back to the Sleepy Science Channel. Tonight, you're shrinking metaphorically. Don't panic down into the microscopic universe humming inside your own body. It's a world where trillions of cells work overtime, mostly without complaining, and each one carries your complete genetic blueprint. Imagine every skin cell, liver cell, and brain cell clutching the same instruction manual like overworked interns, but somehow each one only reads the chapter it needs. Heart cells skip the digestion chapter. Stomach cells don't bother with how to beat rhythmically. Everyone minds their business. Before you get too cozy, go ahead and like the video or subscribe, but only if it genuinely makes your serotonin wiggle. And feel free to drop a comment telling me where you're watching from and what time it is. I love imagining this little sleepy classroom scattered across the globe. Now dim the lights, let the room hum softly, and let's drift together into the quiet chaos of genetics. And here's where things get magical. If you could take the DNA from just one of those cells and stretch it out, it would be about 2 meters long. 2 meters. Meanwhile, it currently fits inside a nucleus so tiny it would lose a staring contest with a dust speck. This is molecular origami at its peak, folding and refolding information tighter than the last t-shirt you packed for vacation. Now scale that up. All the DNA in your entire body, laid end to end, could reach from Earth to the Sun and back multiple times. That's not metaphorical. That's physics. You are basically a cosmic yo-yo of biological information, a walking encyclopedia wrapped in skin, pretending not to be extraordinary while scrolling your phone at 1 a.m. At the heart of all this elegance sits DNA deoxyribonucleic acid, looking like a tiny twisted ladder that spirals toward infinity. The sides are made of sugar and phosphate molecules, a sturdy backbone holding everything together, while the rungs are formed by base pairs that snap together with the accuracy of a celestial jigsaw puzzle. Its design and function rolled into one elegant helix, nature's version of minimalist architecture. Scientists first visualized this double helix back in the 1950s, a revelation so dramatic it might as well have come with theme music. Suddenly, the world understood how genetic information could copy itself, each side of the ladder acting as a template for the next. Simple, elegant, terrifyingly efficient. Under a strong enough microscope, stronger than anything in your kitchen drawer, you'd see the chromatin threads curling and tangling like cosmic noodles inside the nucleus. When you zoom past the limits of light microscopy, the dance of the double helix unfurls. It's like a molecular twist contest. 2 strands entwine in a graceful spiral, reminiscent of a staircase spiraling downward into the depths of heredity. The real magic lies in the genius of just 4 chemical letters: adenine, thymine, guanine, and cytosine. Yup, just a T, G, and C form the entire blueprint of life in a simpler alphabet than most of our texts. Now, here's the real kicker. These bases have their own pairing buddies. Adenine cozies up with thymine, while guanine finds its perfect match in cytosine. This buddy system, discovered through some mind-boggling chemical sleuthing, is the secret sauce behind DNA's self-copying abilities. When the helix unzips, each strand acts as a template, ensuring that the right buddies come together to make identical copies. Magic? More like molecular wizardry. As the 4-letter sequence dances through life, it knows no bounds. A single gene can start as simply as ATGCG and stretch on for thousands. The sequence is paramount to change just 1 letter, and suddenly you might unlock a new trait or maintain the status quo. Some sequences direct the construction of proteins, while others flip switches like light bulbs, deciding when genes get the green light to shine bright or dim down. Speaking of proteins, think of them as the hard-working gnomes bustling within cells. Each gene serves a purpose, like a recipe in a cookbook, instructing the construction of specific proteins essential for life. From the keratin giving your hair that glorious shine to enzymes speeding up life's chemical reactions, proteins are the workhorses hustling behind the scenes. A gene can be a tiny recipe or an epic saga extending thousands of letters long. When a cell needs its superstar protein, it calls in the molecular machinery to read its corresponding gene. The 4-letter code is magically translated into chains of amino acids. Think of them as the building blocks of proteins. Each triplet in the DNA corresponds to one amino acid, like a recipe that guides the sequence and ultimately determines what the protein will do on the grand stage of life. Humans boast approximately 20,000 genes shuffled throughout their cozy genome habitat. While some genes are the all-star household members active in every cell, like the trusty housekeeping genes, others play niche roles, only popping out to perform for their special event. For instance, the insulin gene takes the spotlight mainly in pancreatic cells, while the hemoglobin gene loves its time in developing red blood cells. And now, let's talk chromosomes. They're like the neatly packed boxes of your genetic belongings. Your entire genome, all 3 billion base pairs, is organized into these fabulous structures. Each chromosome is a long DNA strand wrapped around proteins called histones, snugly bundled to protect and organize our precious genetic treasure. In total, humans flaunt 46 chromosomes, or 23 pairs, one from the motherly team and one from dad's side. These chromosomes strut their stuff and are most impressive when a cell is dividing, condensing into those eye-catching X-shaped structures, ready to propagate life's marvelous secrets. When cells aren't in the midst of dividing, chromosomes decide it's time to loosen up and unwind, transforming into a tangled mess of chromatin that occupies the nucleus like confetti at a party. This relaxed state is a boon for the cell's machinery, granting easy access to DNA. It's like having a backstage pass to the gene-clad rock stars, where they read the scripts and copy vital info right when it's needed. Each chromosome is a treasure chest, stuffed with hundreds or even thousands of genes and vast stretches of DNA that, for the most part, act more like stagehands than performers. Now, not all the DNA in those chromosomes is clamoring for the spotlight. Some areas are responsible for regulatory duties, deciding when the genes get their moments to shine. Others play the structural role, making sure the chromosomes keep their shape together and properly separate during cell division. For instance, our largest superstar chromosome, chromosome 1, boasts over 240 million base pairs and thousands of genes. Talk about a genetic heavyweight. On the flip side, the smallest player, chromosome 21, still hosts hundreds of essential genes. Every person's genetic mosaic comes from a perfect blend of mom and dad, giving us a smorgasbord of their DNA. When you were conceived, it was like a game of genetic bingo, snagging 23 chromosomes from each parent to create your unique 46. So here you are, a fab patchwork quilt, stitched together from your folks' ancestral fabrics. But wait, inheritance gets a bit trickier. The chromosomes you inherit are like shuffling a deck of cards. Your parents' chromosomes were just as mixed up and switched during their own genetic exchanges with their parents. In a delightful twist, the process of recombination allows chromosomes to trade segments like the ultimate genetic swap meet. When your parents' eggs and sperm were made, their chromosomes paired up and played switcheroo. So, the chromosome you got from mom isn't identical to anything she inherited from her parents. It's a unique blend, making even siblings distinct despite sharing lineage. Of course, the exception to this randomness is identical twins, who share an identical genetic blueprint from a single fertilized egg that splits in two. Now, these identical twins are like nature's greatest genetic clones. They might share the same DNA sequences, but don't mistake that for uniformity. As they grow, differences emerge, thanks to experiences shaping their personalities. One twin might sport a scar while the other remains unblemished, or one could have a quirky affinity for llamas that the other lacks entirely. And as time rolls on, their DNA may even diverge a smidge due to those pesky random mutations that come from cellular divisions. Despite all these differences, the similarities among identical twins are hard to ignore. They often mirror each other in appearance, and you can bet they're likely to have similar personality traits, talents, and preferences. This makes them the perfect subject for researchers eager to dissect what's driven by genetics versus what's influenced by the great, chaotic world around them. When identical twins, raised apart, continue to enjoy the same obscure hobbies, it leans toward genetics being the guiding force, whereas stark differences could shine a light on the experiential aspects of life. On the other hand, let's give a nod to fraternal twins, who are like any other siblings, just with a special birthday buddy system. They come from two separate eggs fertilized by two separate sperm, the real deal, not duplicates. While they share a womb, these twins are the result of entirely different genetic shuffles, just like the rest of us. Each fraternal twin received its unique deck of chromosomes from their parents, ensuring that even though they share similar DNA, they aren't any more similar than ordinary brothers or sisters. It's genetic roulette at its finest. Fraternal twins are like the surprise party of the genetic world. Sometimes they're indistinguishable, while other times they look like they were mixed up at the hospital. It's all due to sharing about 50% of their DNA, which contributes to a delightful variety of traits. One twin might rock their mom's sparkling blue eyes, while the other flaunts dad's deep brown. Oh, and let's not forget the height difference. One could be strutting down the basketball court while the other is still trying to find their shoes. In the realm of twins, fraternal ones are the divas, with their appearances dictated by the complex dance of genetics. Some families love the twin gig so much that they churn them out through generations, hinting at a mysterious genetic secret. In contrast, identical twins are like the ultimate lottery win, popping up randomly across the globe. Their 1 in a million split from the same embryo doesn't seem to pay homage to genetics. It's more like luck struck. Speaking of genetics, it's quite the tapestry with billions of DNA-based pairs weaving through humanity. Every individual has a unique cocktail of DNA variations, thanks to single nucleotide polymorphisms. Let's call them SNPs for fun. Most of these SNPs just hang out quietly, creating diversity without making a ruckus. But some can stir the pot, like when a single letter changes tailors and enzymes hustle or turns a receptor into a drama queen. These genetic quirks gather momentum over generations, revealing family ties while intertwining our ancestral roots. Each individual's DNA decor not only narrates their story, but also reveals their genealogical gossip. Populations carry their distinct signatures, meaning a group of cousins might share more genetic flair than the neighborhood bowling team. This unique blend of genetic variants is your familial cocktail, passed down through the family tree's colorful branches. Mutations, the lovable misfits of the DNA club. Occasionally, they crash the party when DNA copying goes awry. These changes can arise due to environmental mischief or just because, well, they can. Most of the time, these little rascals are harmless, taking residence in non-coding regions or subtly altering a DNA letter without disturbing the peace in the protein community. Yet fear not. Some mutations flip the script in a heroic way, bestowing survival advantages that might just help their hosts thrive. These golden mutations sneak into the population's dynamic, nudging evolution along its merry way, while others can take a turn for the worse, causing proteins to go rogue or disrupting key genetic processes with unfortunate timing. And let's not forget, each of us carries a bag of tricks called mutations that sprout from our parents' DNA during the origin story of life. Most mutations are benign, just along for the ride. However, a few might take center stage in our traits or health conditions, continuing the genetic narrative that shapes who we are. In the grand scheme, some mutations are just party crashers, causing genetic disorders and reminding us that DNA can be a wild ride. Genetic disorders can often feel like a twisted version of a funky game of telephone. One tiny change can completely alter the message. In the world of genetics, a single base pair mutation can cause conditions like sickle cell anemia, where red blood cells morph into a sickle shape. This transformation triggers a host of painful complications, all thanks to a minuscule mix-up in hemoglobin. It's baffling how such a small change can cause such big trouble. Then there are those genetic disorders that enjoy the thrill of being a chromosome above the crowd. Take Down syndrome, for instance, where having an extra chromosome 21 throws a party, bringing along its pals' developmental quirks and distinct features. On the flip side, we have Turner syndrome, the secretive condition where a female is sporting only one X chromosome instead of the usual pair. Talk about a family reunion gone awry. Chromosomes can really shake up normal development. Not all genetic disorders are knockouts, though. Some are recessive, hiding in the shadows until two carriers decide it's their time to shine. These sneaky mutations are like OK Cup wildcards, only visible when both parents slip in their hidden versions. What's double trouble for one couple might mean cystic fibrosis or Tay-Sachs disease for their child. It's like a genetic lottery that no one wants to win in this case. Spoiler alert, it's serious business. Speaking of winning, let's rewind to Gregor Mendel, a monk with a passion for peas and a knack for inheritance. He cleverly found that certain traits, like flower color, follow the good predictable patterns. Cross a purple flower with a white one, and voila, all purple. But dive deeper, and we discover that when those purple offspring tango, white flowers suddenly crash the party. Mendel's discoveries laid the groundwork for our understanding of genes, turning him into the godfather of genetics. Mendel's simple model might explain widow's peaks and tongue rolling, but buckle up, human traits are a wild genetic cocktail. Unlike Mendel's straightforward P's, traits like eye color, height, and even smarts involve a dizzying array of genes playing tag with various environmental factors. Think of it like an elaborate dance-off where many genes bring their A game while nutrition and life experiences throw in their own funky moves too. So, what about the colorful world of eye color? It turns out it's not just a one-gene wonder. While older theories suggested a simple setup, recent discoveries reveal an intricate interplay among numerous genes that decide our peepers' hues. Our dear friends, the OCA2 and HERC2 genes on chromosome 15, take center stage in this show, influencing melanin production and ultimately the shade of our irises. We've all seen two brown-eyed parents with a blue-eyed kid do a double take, right? The mysterious eye color battle showcases how genetic complexity can surprise us. While brown might reign supreme, sometimes those recessive blue alleles tag along for the ride, revealing the beautiful spectrum of eye colors from chocolate brown to frosty blue. Who knew the genetic recipe was so diverse, creating a living rainbow right before our eyes? Now that's a plot twist worth celebrating. Ah, the joys of genetics. Did you know that even the amount of light your baby is exposed to can influence their eye color? Most infants arrive in the world with charming blue or gray peepers, full of potential. But hold on to your baby booties, because as those first 12 months roll by, melanin kicks into gear. By the time your little one hits the big three, their eye color typically reflects the unique mix of genes gearing up for a captivating reveal. It's like a color-changing magic show. Now, let's talk about height, because who doesn't want to know how tall they're destined to be? Height is a fascinating trait influenced by a multitude of genetic variations, think hundreds, if not thousands. Unlike Mendel's straightforward pea plants, human height is a bit more complicated. It's not as simple as a tall gene and a short gene. Rather, countless variants come together to determine whether you'll snag the last cookie from the top shelf or need a lift to reach it. Research shows that around 80% of our height is influenced by specific genetic variants, making it one of the most heritable traits known to mankind. So if your mom and dad are towering giants, chances are you might skip the kiddie rides and head straight for the roller coasters. But don't forget environmental factors like nutrition. A well-fed kiddo might find themselves stretching past their siblings on the family height chart, even if the DNA looks the same. This brings us to the age-old debate: nature versus nurture. While genes set the stage, they don't dictate the performance. Factors like nutrition, education, and lifestyle play vital supporting roles. Your buddy might have genetic potential for genius, but without that brain food or a little scholarly encouragement, they could fall short. The classic expression, genes load the gun, but the environment pulls the trigger, sums this up perfectly. And guess what? Not all genes work at full throttle. Sure, every single one of your cells contains the same genetic blueprint, but they don't play the same notes. A muscle cell isn't rocking out the same tune as a neuron. This is where gene expression struts in. Depending on which genes are turned on or off, a cell can do its own thing, staying in sync with its specific job in the grand performance of life. Imagine gene expression as a blockbuster movie where certain chapters of that shared genetic script get read differently by different cells. Transcription factors act like directors, calling the shots on which genes take the spotlight. Meanwhile, enhancers and silencers provide added drama, determining whether a gene comes to life or takes a back seat. It's all a spectacular production, one where your genes are the actors, but the environment provides the stage. Through it all, the intriguing dance between genetics and environment continues to unfold. Your journey is a personal blend of choices, experiences, and a sprinkle of luck, crafting a story that's uniquely yours. So whether you're blue-eyed, tall, or bursting with bright ideas, remember, while genes lay the groundwork, you are the author of your life's narrative. DNA is like the master script of life, but its expression levels are fine-tuned by stretches that sometimes act like stage directors often far from where the actual genes reside. This cellular Broadway show is responsive to outside signals. Hormones strutting on stage, nutrients playing supporting roles, and stress providing unexpected plot twists. The precision of this regulation keeps the whole performance in harmony. During development, an encore of gene expression takes the spotlight, directing the formation of heads, limbs, hearts, and brains with meticulous choreography. One tiny mishap in this production could lead to developmental hiccups or even diseases. But when the script is followed correctly, you get the glorious complexity and functionality of a living organism, all from a single genetic source. Talk about a family reunion. Now, let's take things to a new level: epigenetics. This fancy term literally means above genetics, and it represents a cool twist in the genetic saga. Here, chemical modifications to DNA and its associated proteins, the supportive cast, if you will, come into play. These modifications don't change the actual DNA sequence, but are the hot ticket to determining whether a gene gets to shine or gets a starring role in the dark. One of the most popular epigenetic marks is DNA methylation. It's like putting a "do not disturb" sign on a gene, making it inaccessible for the cell machinery to read. Remove that methylation, and you've activated a hidden gem of genetic potential. Let's not forget the unsung heroes known as histones. These proteins are what DNA wins around, and their modifications can either open the curtains on relevant gene regions or close them tight, depending on the play at hand. What's mind-blowing is that epigenetic marks are influenced by what happens in the outside world. Diet, stress, toxins, you name it. Some changes can even be handed down to the next generation, a topic that's still causing coffee shop debates and plenty of active research. Imagine having characteristics passed down from a thrilling life experience. It's both fascinating and a tad controversial. Alright, so what's the bridge between the majestic DNA and the wondrous proteins? Enter RNA, the trusted messenger. While DNA keeps genetic info safely stored, RNA, a close chemical cousin, takes on the task of protein production. When a gene gets the green light, RNA polymerase swoops in, reading the DNA and crafting a complementary RNA copy. This messenger RNA, or mRNA, carries instructions from the nucleus to the bustling cytoplasm, where the real magic happens. It's like sending out invitations to a party. The ribosomes are ready to respond. Once in the cytoplasm, ribosomes take the script from the mRNA and translate the genetic code into a sequence of amino acids. Think of these ribosomes as chefs in a busy kitchen, whipping up delicious proteins, adding amino acids at a rapid pace of several per second. And just to up the ante, multiple ribosomes can be reading the same mRNA at once, cranking out countless copies of a protein from a single script. But RNA doesn't just play a one-note tune. It's far more versatile than its DNA counterpart. While some RNA types serve as the star performers, others hang back and take on regulatory roles. MicroRNAs, for instance, like to poke at mRNAs, making sure they don't get translated too much. Meanwhile, ribosomal RNA kicks it behind the scenes, forming the ribosome structures themselves. Not to mention transfer RNA, which joyously delivers amino acids to the ribosome for that perfect protein synthesis moment. In the grand finale, proteins are the rock stars of your cells, performing a myriad of functions that keep your body ticking. Just think of all that coordination, a vibrant dance of DNA, RNA, and proteins, orchestrated in a spectacular genetic performance that ensures life is anything but dull. The beauty of it all is that through these layers of regulation and expression, life becomes the fascinatingly complex saga that we get to live every day. Proteins are the unsung heroes of life, acting as the builders and the workers in our biological frameworks. Each protein is unique, designed with a specific shape tailored for a specific task. Enzymes, for instance, are the speedsters of the cellular world, hastening chemical reactions that would otherwise take eons. Whether it's munching through our meals with digestive enzymes or tapping energy with metabolic ones, proteins truly get the job done. In the realm of structure, proteins are the architectural superheroes. Collagen is like the tough guy on the block, forming fibers that provide support in skin and bones, while keratin hardens up as it creates hair and nails. Ever seen muscles flex? That's the dynamic duo, actin and myosin, sliding around like dancers in a grand performance, showing off their powerful contractions. It's a wild protein party that keeps everything in shape. But wait, there's more to these protein characters. Some take on the critical roles of messengers and receptors. Insulin, for example, is the friendly signal that tells cells to gobble up glucose like it's candy. Then there are antibodies, the vigilant guards that sniff out invaders. Receptor proteins are the sensitive types, picking up signals from hormones and neurotransmitters and alerting the cell to respond like a well-oiled communication system that's always on alert. Now, let's dig into the chic world of protein folding. A protein begins its life as a simple chain of amino acids, but it quickly dives into a unique three-dimensional shape that's crucial for its function. Imagine it like dressing up for a party. Some proteins need to fit together like a lock and key to work their magic, while others must match their comrades exactly. If a protein gets tangled or crumpled in the wrong way, it might not only flop but could also become a liability. How do proteins avoid tragic wardrobe malfunctions? Enter chaperones, the personal trainers of the protein world. These specialized proteins swoop in to assist newcomers, ensuring they fold correctly and don't get tangled up. But if misfolding does occur, other robust systems are on standby to recognize and toss out the troublemakers. When these systems fail, it can lead to a mess of diseases like the plaque buildup in Alzheimer's that nobody invited. The quest for perfect protein folding is like the ultimate biological puzzle. Yet, predicting how an amino acid sequence will curl and twist is no cakewalk. There's a glimmer of hope, though advances in artificial intelligence are stepping in to crack the code. If we can figure this out, it could change the game in medicine, enabling us to design proteins for specific purposes or potentially fix diseases caused by misfolded proteins. It turns out the genetic code, the instructions for making proteins, is nearly identical across all forms of life. This fascinating discovery reveals that the same DNA triplet responsible for a specific amino acid in a human also does the same in a bacterium or a whale. With only a handful of exceptions, nature seems to play by the same genetic rulebook. This shared coding hints at a shared ancestry that connects all living things, showcasing that evolution has been a consistent, decisive force over billions of years. The beauty of this universality is that it simplifies genetic engineering. Scientists have cleverly harnessed this common language, enabling them to insert human genes into bacteria that manufacture proteins, like insulin, efficiently. This opens the door to monumental advancements in medicine and biotechnology, showing how proteins, with their universal language and roles, are at the heart of life itself. In a world where bacteria have taken a crash course in human genetic engineering, these tiny powerhouses are producing insulin for diabetics in fermentation vats. Who knew our shared genetic legacy could be so beneficial? It's like having a few microscopic friends that know how to whip up essential life-saving hormones, thanks to the fact that every living organism shares some genetic material. So, you may have more in common with a banana than you ever imagined. Speaking of shared genes, you'd be surprised to hear just how much we have in common with other species. For example, humans and fruit flies share a staggering 60% of their DNA. This means the next time you swat a fly, remember they might be genetically related to you in some peculiar way. From flies to mice, 90% similarity, and even to dogs, 85%. It's clear that many of the fundamental genes for metabolism and development are ancient and well preserved across the evolutionary tree. Who knew our distant relatives had such a talent for survival? Interestingly, even plants aren't off the genetic hook. Every living thing needs to convert energy, build proteins, and fix damage, which points to our shared ancestry. While each species varies in its unique adaptations and quirks, the underlying foundation of life, those essential genes we all share, remains constant. Think of it as a gigantic family reunion with all sorts of relatives bringing their own unique flavors to the table, but still sharing the same genetic recipe. Evolution isn't just a grand tale written in the stars. It's also a slow, meticulous process shaping our genomes over millions of years. The genome is not a rigid structure. It's more like a party that evolves with time through mutations, natural selection, and gaudy genetic dances. If a mutation enhances survival, it's like winning the genetic lottery. It gets passed down through generations. Meanwhile, less favorable mutations often get voted off the island, ensuring that only the fittest variations thrive. Within a population brimming with genetic diversity, natural selection kicks into action, ensuring that some lucky individuals are better suited to dodge predators, resist pesky diseases, or hunt down dinner. These advantageous traits spread like wildfire through the gene pool over time, illustrating just how iterative evolution can be. It's all about survival of the fittest, but let's not forget those neutral mutations that just wander in and out of populations without a care in the world. Diving into the human genome is like uncovering layers of a fascinating historical manuscript. With every comparison to our primate cousins, we get a glimpse into what changed during our evolutionary journey. Those clever changes that affected our brain size and cognitive skills took center stage. Evolution had a lot to say about our mental bandwidth. It's a narrative of growth and adaptation woven seamlessly into our DNA, an exquisite palimpsest of life's history. Despite showing a staggering 99% genetic similarity, we are all wonderfully unique. Any two random humans from across the globe will still share a substantial chunk of identical DNA. Yet, the differences which paint the charmingly diverse picture of humanity actually arise from less than a mere 1% of our genetic makeup. Our recent common ancestry, originating in Africa a mere 300,000 years ago, has much to do with this striking similarity that binds us all in a bizarre, intricate genetic web. It's like being part of a special club where we're all relatives is the unifying motto. Diversity among human beings manifests in various forms, from skin color and hair texture to the unique shapes of our faces. Yet, beneath these visible traits lies an astonishing truth. The majority of our genetic makeup is nearly identical across all populations. This genetic unity shatters the myth of distinct human races, revealing that we are one species with only minor discrepancies. It's a humbling reminder that deep down, we are all more alike than different. Ancient DNA offers a thrilling glimpse into our evolutionary saga. While DNA has a short lifespan and typically degrades within a few thousand years, it can survive in pristine conditions for eons. Scientists have successfully extracted genetic material from ancient bones and teeth, piecing together our ancestral tales. Some of these revelations are downright juicy. For instance, Neanderthals interbred with modern humans, leaving a curious legacy of 1 to 2% Neanderthal DNA in many of today's folks of European and Asian descent. Talk about a wild family reunion. But wait, there's more. Enter the Denisovans, another intriguing group of ancient humans mostly known from scant remains in Siberia. This bunch also had a hand in shaping the gene pool of modern humans, especially in Southeast Asia and Oceania. Ancient DNA reveals a thrilling narrative. Migration patterns, cultural exchanges, and historical events have all woven together the genetic tapestry of humanity. Imagine it as a fantastic family tree crafted not just from photos but from the very essence of our genes. The emerging field of paleogenomics is captivating as it melds genetics with archaeology and anthropology. This combination is akin to a time machine, offering complex insights into our past that mere artifacts could never unveil. Each ancient genome acts as an emissary from bygone eras, echoing the lives once lived, while reminding us that our shared history is anything but straightforward. It's a rich saga waiting to be uncovered, one tiny fragment at a time. Now, shifting gears a bit, let's discuss mitochondria, those tiny powerhouses buzzing around in our cells. Alongside the nucleus, these organelles work tirelessly to convert nutrients into energy. Interestingly, mitochondria pack their own punch in the DNA department as well. Mitochondrial DNA, mtDNA, is just a fraction the size of nuclear DNA, yet it possesses a significant backstory. These little power plants were once independent bacteria that hitched a ride in ancient cells, establishing a mutually beneficial relationship instead of facing digestion. What's even more fascinating is how mtDNA is inherited. Unlike nuclear DNA, which scrambles and mixes from both parents, you inherit all your mitochondrial genes from your mother. Yes, that means your mtDNA is a direct line stretching back generations, identical to your mother's and her mother's, leading all the way back to an ancient matriarch. This unique inheritance pattern offers researchers a fantastic tool for tracing maternal ancestry and mapping out human migration through the ages. Thanks to this miraculous mtDNA scheme, scientists have built an elaborate family tree of humanity, tracing back to a woman affectionately dubbed mitochondrial Eve, who roamed Africa around 150,000 to 200,000 years ago. While she wasn't the only lady at the time, she's the one whose lineage continues today. So, every single person alive carries a fragment of her genetic legacy, proving that we are all distantly related. What an unbelievable cosmic connection we share. As time marched on, our ancestors fluttered like pollen through the winds of Africa, splintering off worldwide like a genetic confetti explosion. These adventurous souls racked up mutations along the way, creating unique maternal lineages known as haplogroups. These groups tell tales of ancient migrations, mapping out where our forgotten relatives roamed. Tracing your maternal haplogroup is like reading a historical map, revealing the epic journey of your forebears from Africa to every corner of the globe, through frigid ice ages and fluctuating climates, until they settled down to form the very foundations of your family tree today. On the flip side, we have the Y chromosome, the unsung hero for tracing paternal ancestry. Only the dudes get to wield this iconic marker passed down from father to son. It's a nifty little trick that allows us to uncover our paternal lines in a way quite similar to mitochondrial DNA. Yet, just like its female counterpart, the Y chromosome doesn't dabble much in recombination, ensuring that lineages remain intact and recognizable through generations. Watch out, mutations they're accumulating and creating distinct Y chromosomal haplogroups, mapping out a journey that shines a light on the paths taken by ancient men. And speaking of ancient men, meet our common male ancestor, living it up in Africa between 200,000 and 300,000 years ago. Sure, he wasn't the only man kicking around, but his Y chromosome lineage had some serious staying power. Fast forward to now, and the Y chromosome is your trusty guide to the tales of historical migrations, the blending of cultures, and the influence of legendary figures like Genghis Khan and the Vikings. This genetic panorama paints a vibrant picture of how human populations scattered across continents and shaped our communal past. Diving deeper into genes, we encounter the fascinating world of dominance and recessiveness. It's the genetic equivalent of musical chairs. When two different versions of a gene come into play, only one takes the spotlight. Sometimes, one allele hogs the limelight by being dominant, while the other is a timid, recessive number lurking in the shadows. Take Huntington's disease, for example. Just a single mutated copy can unleash havoc. But wait, don't fret just yet. Dominant disorders tend to be pretty rare since they usually lower the chances of passing the mutation downstream. Recessive disorders, however, are master escapologists, sneaking through family lines like ninjas without causing immediate havoc. Carriers can maintain their healthy statuses, but when two of them decide to make mini-me versions of themselves, there's a 25% chance their child will inherit both mutated copies, turning the tides. Diseases like cystic fibrosis, sickle cell anemia, and Tay-Sachs are all in this club, reminding us all about the importance of understanding our genetic lottery tickets. But let's not get lost in the worry. Some mutations are like superheroes in disguise. Not every genetic twist is a villain waiting to wreak havoc. A mutation in the CCR5 gene can grant you resistance to HIV, acting as a stronghold against the virus. This mutation is like a secret weapon, especially prevalent in European populations, possibly because it offered a buffer during historic plague pandemics. Similarly, the sickle cell trait serves a dual purpose. Just one copy protects against malaria without sending the carrier down a path of disease. Talk about a genetic win-win. In the end, understanding our genetic legacy isn't merely a family history project. It's an investigation into our unique human story, woven with laughter, heartache, and sheer survival. As we piece together the intricate tapestry of our ancestry, we discover that while we're all on different paths, we share a common bond that links us all both through triumphs and tribulations. So next time you ponder where you come from, remember the wild, winding journey of those who came before you and how it perfectly frames the beautiful mosaic of humanity we inhabit today. In the world of genetics, some variations are like the cool club that everyone wants to be part of, like the sickle cell allele. Found in regions plagued by malaria, This particular gem gives a significant survival advantage to those who carry just one copy, while two copies create a cocktail of health issues. It's like winning a game of roulette where the odds are ever in your favor if you're not doubling down on that risky bet. Instead of fading away, the sickle cell trait remains in the gene pool, showing us that survival can be a funky dance between adaptation and sheer weirdness. Then there's the lactose tolerance saga, an epic tale of humankind's love affair with milk. Most adult mammals hit a milk wall after weaning, but our dairy-loving ancestors decided to hang onto that sweet, creamy goodness. Mutations arose like superheroes, keeping the lactase enzyme active and thus allowing those adults to enjoy milk well into adulthood. Thanks to these mutations, Europeans, some Africans, and people in the Middle East thrive on cheese, yogurt, and lattes, while the rest of us are left eyeing that ice cream with a disappointed stomach. Talk about a dairy dilemma. Blood type, truly the ultimate party theme of human genetics, introduces us to a colorful array of identities: A, B, AB, and O. This whole setup is encoded in the genes on chromosome 9, which additionally control what sugar toppings adorn your red blood cells. If you land on type A, you've got one flavor, while type B brings another to the mix. Type O is like that friend who shows up to the party but forgets to bring the snacks no sugar added here. The combinations only add to the fun, leading to a delightful blood typing classification battle where A and B are the co-stars, while O takes a casual backseat. But wait, blood type isn't just for show. It's crucial during transfusions, and if you get a surprise blood type that your body doesn't recognize, things can get messy. Picture your immune system as the bouncer at a club. If it doesn't recognize the attendees, the antibodies, it can kick them out with a vengeance. Fortunately, type O serves as the universal donor, slipping in unnoticed at the party and saving the day in emergencies. On the flip side, AB is the VIP accepting all types. So, who knew our blood could spark such drama? The saga continues as we explore how blood type can also confirm parentage. Think of it as the family reunion of genetics. If both parents have type O blood, all offspring are guaranteed to rock the O blood type. However, throw an AB parent into the mix, and the O traits disappear faster than pizza at a party. That's right. Despite DNA testing being the new hotshot in the paternity world, blood type remains a trusty sidekick. As if that's not enough, we uncover the intriguing Rh factor. Blood types aren't just about A, B, AB, or O. They also come with a side of positivity or negativity. Rh positive is the popular kid at recess. Around 85% of people fall into this category. However, the plot thickens during pregnancy. If an Rh negative mom is baking an Rh positive baby, her immune system might start swinging punches, thinking the baby's cells are intruders. Modern medicine comes to the rescue with Rh immunoglobulin, a necessary shot for those Rh negative moms, helping them avoid a showdown with their own baby's blood cells during subsequent pregnancies. It's a brilliant twist of fate, weaving together genetics and immunology in a way that protects future generations. And just like that, Rh positive becomes the dominant player, ensuring that the dance of life continues on without missing a beat. You know, being Rh positive is a bit like having a VIP pass in the world of genetics. Just one copy of the Rh gene and you're in. On the flip side, Rh negative folks are like the exclusive club members with two copies of a non-functional version, meaning they can only produce Rh negative children. Meanwhile, those Rh positive parents can be tricky if they're both hiding the recessive gene. Their child might just be unlucky enough to join the Rh negative club. Who knew genetics could feel like such a high-stakes game? Now, let's talk about the wonders of genetic testing. Thanks to advances in DNA sequencing, it's as easy as sending in a saliva sample and cashing out on your ancestry report. For a slight financial pinch, companies will lay out your lineage and reveal your quirks, from your love of kale to your potential penchant for terrible karaoke. But wait, there's more. Medical testing can point out potential diseases waiting in the wings, like breast or colon cancer. Yikes. With this kind of intel, you can make informed choices about your health. Talk about living with your eyes wide open. Imagine finding out you have a mutation in the illustrious BRCA1 or BRCA2 genes. This isn't just trivia. It means your chances of developing breast and ovarian cancer have skyrocketed. With this juicy information in hand, you could step up screenings, think about preventive surgeries, or even ditch that extra slice of cake. The beauty of genetic testing is the power it gives you. It can save lives and help you dodge health bullets before they're fired. But before you get too excited, it's important to bear in mind that genetics is a wild ride full of probabilities. Here's the kicker, though: having a genetic variant doesn't mean your fate is sealed. It's like owning a treasure map that may or may not lead you to gold. There are too many spices in this recipe. Environment, lifestyle, and just plain old luck can all shake things up. And then there's the ethical quagmire. Who gets to see your genetic secrets? Should insurance companies or bosses have a sneak peek? Although many laws now protect your precious data, the specter of genetic discrimination still looms large. And if that's not enough, there's the curious case of incidental findings. Imagine going in to check for the pesky gene that makes your hair frizz, only to find out you're at risk for some rare condition that sounds like a character in a sci-fi flick. It's a slippery slope that society is still navigating. Suddenly, genetic testing feels less like a personal journey and more like a thrilling game show, except you didn't sign up for half the surprises popping up. Now, gene therapy, my friends, is the ambitious superhero of genetics. The aim here? Fix those pesky, faulty genes that kickstart diseases in the first place. The idea is as sweet as pie. Introduce a functional gene into the body and let it work its magic. However, like any great superhero tale, the journey has been fraught with challenges. Early trials weren't exactly smooth sailing, with patients experiencing immune reactions and even cancer from rogue gene insertions. Oof! Safety concerns put a pause on this promising hero. But hold on to your lab coats, because recent breakthroughs have sent gene therapy soaring. Think about kids cured of the bubble boy disease and those regaining their sight from inherited blindness. It's like magic. Treatments for sickle cell and beta thalassemia are gaining momentum too. With price tags still high and availability limited, gene therapy feels exclusive, yet it's evolving quickly. New players like the CRISPR technology are taking the center stage now, bringing precision to genetic corrections that earlier methods could only dream of. CRISPR is the real game changer here, giving scientists the ability to slice and dice DNA with pinpoint accuracy. Picture it as molecular scissors guided by a sophisticated GPS system. This is all thanks to bacteria who knew they could be such brilliant problem solvers. With a short RNA directing the CRISPR-Cas9 protein to target specific genome locations, it snips the DNA, letting scientists play God with how those cuts are repaired, whether by deleting undesirable sequences, fixing mutations, or introducing shiny new genes. Who knew genetics could be this exciting? The advancements in gene editing technology, specifically CRISPR, are taking the scientific world by storm. This revolutionary technique is not only faster and cheaper than those old-school methods, but also packs more precision in its toolbox. Researchers are using CRISPR to study genes in organisms ranging from pesky bacteria to us, human folks. It's helping to craft disease models and dream up new therapies for conditions like sickle cell disease, muscular dystrophy, and some cancers. Who knew science could be so cool? But here's the twist: along with the thrilling potential comes a heap of ethical questions. If we can tweak human embryos, should we really do it? What about those superhuman enhancements instead of just curing diseases? Suddenly, we find ourselves standing at the crossroads of progress and conscience, facing the potential to rewrite the entire human genome. It's kind of like playing God, but with medical textbooks, which is both exhilarating and a tad nerve-wracking. Then there's the whole cloning saga, which sounds like it's straight out of a sci-fi flick, but it's totally real. Dolly the sheep is the celebrity of cloning fame, born back in 1996. Imagine this. Scientists took a cell from an adult sheep's udder, yanked its nucleus out, and popped it into an egg cell. Afterwards, they gave that egg a nudge to grow, and voila, Dolly arrived, a genetic twin of the original sheep. Dolly showed the world that adult cells have the original genome tucked away and can be reprogrammed to create living organisms. Cloning isn't just a woolly wonder. It's been whimsically applied to a variety of animals from cows to cats. Researchers clone for various reasons: preserving the genetics of prized livestock, studying diseases, or just peeling back the layers of basic biology. Now, the idea of human cloning, Not so much. It's a controversial topic that has been halted in many places due to significant technical hurdles and hefty ethical dilemmas. Still, the concept of reviving the past by resetting that genetic blueprint is maddeningly fascinating. Now, let's not forget about nature's version of cloning. Picture identical twins, nature's original cloning experiment. These twins emerge from a single fertilized egg, which splits into two separate parts after a few divisions. They may share the same genetic material, but they march to the beat of their own drums. Each twin flourishes independently, bringing unique personality traits, experiences, and even fingerprints to the table. It's a wild scientific wonder that shows nature can be just as quirky and captivating as any lab. While identical twins share a remarkable genetic bond, they aren't clones in the strictest sense. Over time, their experiences and environmental factors will shape them differently, leading to slight divergences in their genomes. All of this makes twins perfect subjects for researchers as they help unravel the tussle between genes and environment in shaping human traits. Who knew being a twin came with such scientific street cred? And then we have stem cells, the superheroes of the cellular realm. These unspecialized marvels have yet to decide if they want to become skin, nerve, or something entirely different. Embryonic stem cells are like the untamed wildlings of the cell world, ready to transform into any cell type. But as they grow older, adult stem cells become a little pickier, sticking to specific duties like replenishing blood or skin. They may not have unlimited potential like their embryonic counterparts, but they still play a crucial role in healing and regeneration. Adult stem cells are the unsung heroes of our biology, tirelessly maintaining and repairing our tissues throughout our lives. Recently, scientists threw a wild twist into the mix by reprogramming these adult cells into an embryonic-like state, giving rise to the cool kids of the cell world: induced pluripotent stem cells, or iPSCs. By using specific genes, they can morph a humble skin cell into a versatile stem cell that can potentially transform into numerous cell types, all while gracefully sidestepping ethical dilemmas involving embryos. Thanks to this brilliant breakthrough, the future of stem cell research looks bright and promising. Imagine a world where damaged hearts are healed, spinal cord injuries are a thing of the past, Parkinson's disease is a distant memory, and we can even grow replacement organs. As scientists decode the complex genetic programs that govern stem cell differentiation, they're peeling back the layers of how genes choreograph a magnificent dance of development within our bodies. The rhythm of life unfolds astonishingly, beginning with that tiny fertilized egg. From that singular egg, nature goes on to orchestrate an entire organism complete with trillions of cells expertly organized into a symphony of organs and tissues. It's like the universe's most intricate puzzle, and everything follows an incredible sequence of genetic activation, unfolding in impeccable timing. Early genes kick off the process by neatly sectioning off the embryo, while later genes step in to define what forms where. Eventually, the finicky details are honed as master control genes, like the legendary Hox genes, choreograph where limbs sprout and how everything is arranged. These Hox genes are ancient architects, guiding the foundational blueprints for body parts. When these genes go haywire, you might end up with fly legs strutting where antennae should be. But don't be fooled. Development isn't a rigid, mindless process. Oh no, the environment plays a vital role too. Nutrition, temperature, hormones, and even your last meal can pivot the way genes express themselves, ensuring each organism perfectly adapts to its surroundings. Now, speaking of genomes, they're not just a spotlight on protein coding sequences. It turns out that genes are merely the star players, making up less than 2% of our entire genetic library. What about the rest? For decades, scientists thought most of it was evolutionary throwaway, but the plot twist is here. Much of this so-called junk DNA actually has purpose. Non-coding regions spring into action by acting as intricate switches controlling gene regulation, slyly managing when and where our genes decide to shine. Among these regulatory goodies are enhancers and silencers, which work behind the scenes to orchestrate the symphony of gene expression. Enhancers can be far removed from the genes they're regulating, creating a complex web of interactions. As if that weren't enough, Some non-coding sequences go beyond regulation, producing RNA molecules that play vital roles in various biological processes. Who knew our DNA had such a zest for multitasking? Let's not forget about the fun, yet infamous transposons, those mischievous little jumping genes. Scattered like confetti throughout our DNA, they have a tendency to bounce around, changing their positions and keeping things interesting. These transposable elements cut themselves out and pop back into new spots, adding an element of surprise to the evolutionary game. With all these quirks and mysteries, it's clear our genome is a treasure trove waiting to tell its story, one that even includes its quirkiest drafts that might someday become part of evolution's unfolding narrative. Imagine a world where genes copy themselves like those pesky photocopiers at the office, constantly churning out duplicates while leaving the original untouched. Around half of our human genome is made up of these little rebels called transposons or jumping genes. Some of them are ancient fossils, dried up by the passage of time, while others are still spry, leaping around and causing a ruckus by disrupting genes or altering regulations. Sure, most of their endeavors are harmless or neutral, but occasionally they throw a surprise party in the genetic structure that yields unexpected benefits. Back in the 1940s, the genius Barbara McClintock went on a wild genetic adventure discovering transposons in corn, proving that DNA is dynamic, not some lifeless scroll. Took everyone a hot minute to appreciate her groundbreaking work, but eventually, she snagged the Nobel Prize. Apparently, the discovery that our genomes are like hyperactive kids on a sugar high was too advanced for its time. Transposons, it turns out, are the unsung heroes of evolution, creating delightful chaos that drives genetic variation across every nook and cranny of life. Speaking of chaos, let's dive into the sheer size of genomes. The human genome boasts a whopping 3 billion base pairs. Sounds impressive, right? But guess what? That's tiny compared to the lungfish, whose genome sprawls over a mind-boggling 40 billion base pairs. Don't even get me started on the Paris japonica plant, flaunting 150 billion base pairs. But fear not, more DNA doesn't necessarily mean more complexity. In fact, some single-celled amoebas have more expansive genomes than mammals. It's a wild ride and indicates that a genome bloated with non-coding DNA is more common than you might think. Hold on to your hats, because genome size is just the tip of the iceberg. Take bacteria, for instance. Their genomes can be as minimal as a few million base pairs, practically pocket change in the genomic world. Symbiotic bacteria can be even more stunningly efficient, cutting down to less than 200,000 base pairs. This snack-sized genome means these wee critters have to hitch a ride on their hosts for many functions, reducing the need for extra genetic baggage. Meanwhile, organisms with larger genomes seem to take the leisurely route, having larger cells and slower metabolisms. Now let's talk chromosomes, those handy-dandy packages of DNA. Humans strut around with 46 chromosomes, while dogs flaunt 78, and fruit flies are basically minimalists with just 8. Just picture it. The single-celled paramecium with dozens of them, and a fern called Glossum reticulatum that holds the ridiculous record with over 1,200. Evolution has a way of throwing up its hands and mixing chromosomes up, sometimes reducing their count or splitting them like a pair of old jeans. The difference between our cozy chromosome count and that of our great ape friends is illuminating. Humans rock 46 chromosomes, while our primate pals like chimps and gorillas boast 48. The mystery lies within one specific chromosome that merges two in humans, separating us from our ape cousins. But remember, chromosome numbers aren't a one-size-fits-all deal. What truly matters is how DNA is organized, and the total genome size bigger doesn't always mean better. Some organisms go above and beyond with their chromosome sets, while we typically enjoy 2, thanks mom and dad. Some plants like wheat strut around as hexaploids with 6 sets. Strawberries take this trend a step further, flaunting 8 sets like they're on a fashion runway. The methods of achieving this are often a wild mix of cell division errors or cross-species hybridization. So while we humans keep our chromosomes neat and tidy, chromosomes in the wild can turn into a flamboyant party. Polyploidy. It's like nature's way of saying, let's have a multiplication party. Sometimes organisms just can't resist adding extra sets of chromosomes when they reproduce. Imagine two different species getting together and bam! They double their chromosomes, creating a whole new polyploid species. While this might sound like a recipe for chaos in the animal kingdom, it's a welcome twist in the world of plants, where polyploidy is a superstar, fueling evolution and agriculture. Plants have hit the genetic jackpot with their extra chromosome sets. These polyploid crops often bear larger fruits and seeds, making them the rock stars of the farming world. Who wouldn't want a watermelon the size of a small child? In animals, though, polyploidy is like that rare, elusive creature that can't stick around. It usually leads to developmental hiccups and is confined to specific tissues. Yet, some fish and amphibians manage to pull off the trick naturally, proving that when it comes to chromosomes, the animal kingdom is just a bit more conservative. Speaking of chromosomes, let's dive into the wild world of sex determination. In humans and most mammals, it's a straightforward XY game. Females flaunt 2X chromosomes, while males rock 1X and 1Y. Ever heard of the SRY gene? It's like the VIP pass for testes development, so without a working Y chromosome, the party goes female. But wait, there's a plot twist. Birds like to keep things spicy with a ZW system, and in some reptiles, searing heat or cool breezes during egg incubation decides the sex. Forget gender reveal parties. Nature's got a mad scientist vibe going. Now, not all creatures fit neatly into the chromosome box. Some have no sex chromosomes at all, and a few even switch genders like fashion accessories depending on social cues. Talk about keeping everyone guessing. It seems that evolution is throwing out all the rulebooks on determining sex, wonderfully diversifying reproduction strategies in the process. Let's not forget about the X chromosome. It's the overachiever of sex chromosomes, housing over 1,000 essential genes. With females carrying 2XS and males brandishing only 1, nature's got a balance issue on its hands. Enter X inactivation, where females randomly silence one X chromosome in each cell, creating a dazzling mosaic of gene expression. It's like a genetic game of hide and seek. A prime example of this genetic artistry is our beloved calico cat. With one X carrying the orange fur gene and the other black, she becomes a walking canvas of orange and black patches, all thanks to X inactivation. Meanwhile, our male feline friends can only dream of such intricate fur patterns unless they happen to have a rare chromosome abnormality. Mother Nature's got a quirky sense of humor. But wait, there's a serious side to this chromosome caper. X inactivation has its say in X-linked genetic disorders too. Ladies with one normal X and one mutant usually skate through life with only mild symptoms because half their cells rely on the healthy X. Poor males, however, are caught in a bind with no backup plan, experiencing full brunt when the mutated X comes into play. This imbalance is why conditions like colorblindness and hemophilia tend to target the boy squad much more often, landing them in the genetic limelight. The Y chromosome may be pint-sized, but it holds its ground with some serious power. This little guy is indeed quite the underdog, boasting fewer than 100 genes that barely fill its tiny frame. Over time, it has shed quite a few of its genes, eventually losing those that were once essential. You'd think that it might vanish into thin air, but surprise! That little Y managed to pull a Houdini act and has stabilized, all while playing a crucial role in determining male sex. Enter the SRY gene, the superhero of the Y chromosome world. This tiny gene packs a big punch. It sets off a domino effect that morphs an embryo into a male. If SRY is missing, even a Y-bearing embryo will develop a female anatomy. Talk about a plot twist. Just one unassuming gene kicks off the construction of male anatomy, shining a light on how the Y chromosome guides our biological destinies. But wait, there's more. The Y chromosome is also the go-to for some essential sperm production genes. If the Y has mutations or deletions, it can spell trouble, leading to male infertility. What's more, since it doesn't mix and mingle like other chromosomes, it's passed down practically unchanged from father to son. It's like a family heirloom, only, with a bit of genetic flair, making it a handy tool for tracing paternal lineages through the ages. Now, let's dive into the wild world of sex-linked traits. If you're always fumbling with colors, it might be because of that sneaky X chromosome. Males, having just one X, are susceptible to X-linked traits like color blindness. About 8% of guys are colorblind, while the odds are drastically lower for females. If a lady wants to sport a similar color challenge, she needs to inherit the mutation from both X chromosomes, a tall order. And speaking of dramatic genetics, meet hemophilia, another pesky X-linked trait. This disorder, which can turn bleeding into a dramatic performance, made its way through royal bloodlines thanks to none other than Queen Victoria. As her descendants married into other dynasties, they inadvertently spread the mutant gene. The saga of royal hemophilia showcases how X-linked traits can wave hello across generations. Moving on to the rare gems of Y-linked traits, these are indeed few and far between. With the Y chromosome sporting a limited number of genes, Y-linked traits are passed exclusively from fathers to their sons, no daughters allowed. This exclusive club includes some types of male infertility and minor characteristics that basically make you think those traits must run in the family. Genes are like those friends who can't sit still. They take part in a genetic version of musical chairs with recombination. During sperm and egg formation, chromosomes line up, swap segments, and create a patchwork of genetic heritage. This chaotic shuffle means that each sibling isn't a clone, even if they share the same parents, as they sprout unique combinations of their ancestors' genes. Finally, let's talk about linkage mapping, the ancient GPS for geneticists before the era of DNA sequencing took over. This clever technique relied on observing which traits frequently appeared together. If you regularly saw certain traits passed down like family gossip, it hinted that their respective genes were hanging out close together on the same chromosome. The tangled web of genetic relationships continues to reveal fascinating insights into heredity, leaving scientists and curious minds cheering in wonder. Researchers delving into inheritance patterns and families embarked on the epic quest of constructing linkage maps, the ancient treasure maps of the genetic world. Creating these scientific marvels was no walk in the park. It required analyzing a treasure trove of data spanning multiple generations and numerous families. These maps became vital for tracking down disease-causing genes. By pinpointing regions of chromosomes consistently inherited with specific disorders, scientists could zero in on the responsible culprits lurking in our DNA. Take Huntington's disease, for instance. In the legendary 1980s, before our genome had even been sequenced, scientists engaged in some serious sleuthing. They scrutinized large families grappling with Huntington's, searching for genetics markers that played hide and seek with the illness. Ultimately, they traced this sly gene to the short arm of chromosome 4, a significant breakthrough that set the stage for later identifying mutations, like the notorious CAG repeat expansion in the HTT gene. Flash forward to the 21st century, where whole genome sequencing has become the shiny new toy in genetic research. With the ability to read the entire sequence, geneticists can now identify mutations directly without the painstaking linkage mapping, though we owe a tip of the hat to the groundwork laid by those early researchers. While linkage studies aren't as glamorous as they once were, they still serve a purpose in certain genetic quests and population genetics studies, reminding us of their historical significance. Speaking of populations, let's dive into the wild world of population genetics. This discipline is like a detective story of how gene frequencies change over time, weaving genetics into the rich tapestry of evolution. It poses riveting questions about diversity, selection, genetic drift, and migration. Within any given population, various alleles can coexist, some like rock stars, and others quietly lurking in the shadows. The frequencies of these alleles are like a dance, constantly shifting under the influence of natural selection, drift, and mutation. The dynamics of these forces shape the genetic carnival within and between populations, making high genetic diversity essential for adaptability. A population festooned with diverse alleles boasts better chances of survival against diseases or environmental upheavals. On the flip side, low diversity can leave a population vulnerable, making the work of conservationists crucial in preserving endangered species and maintaining their genetic diversity. Enter the fabulous world of the Hardy-Weinberg principle, the life of the population genetics party. This principle predicts that allele frequencies should remain steady from generation to generation provided certain ideal conditions are met. Spoiler alert, they rarely are. If a population strays from these conditions, evolution is likely knocking at the door. This helps geneticists detect the forces at play, like mutations tossing in new variants or selection favoring particular alleles. Not only that, but the Hardy-Weinberg principle serves as a handy mathematical tool for calculating carrier frequencies of recessive disorders. By understanding how common a disease is, geneticists can estimate the number of individuals carrying hidden recessive alleles. This has profound implications for genetic counseling and public health. Ultimately, this principle teaches us an astonishing lesson. Without the relentless push of evolutionary forces, rare alleles can hang around for generations, patiently biding their time for conditions that might shift in their favor. And let's not forget about genetic drift, the unpredictable wild card that playfully alters allele frequencies, especially in small populations. In large populations, it's like being on a giant cruise ship. The random waves just rock you gently, and everyone gets well adjusted. But in small populations, picture a dinghy tossed around by wild swells where one out-of-control wave could tip the balance. This is genetic drift at play. Imagine two alleles hanging out at equal frequency, and then by sheer happenstance, the A allele becomes the life of the party, leaving the poor B allele stranded on shore, eventually becoming extinct. It's as though luck is throwing a party, and the winners get to breed while the losers fade into obscurity. Bottlenecks amplify this genetic chaos. Picture a frantic game of dodgeball where only a handful of kids survive a cruel reduction in player count. They emerge from the fray carrying only a small, random selection of the original team's diversity. It's as if they're holding a box of assorted chocolates but only feature a few sad, leftover ones. These lucky survivors then become the genetic foundation of a rebounding population. Meanwhile, the former variety is tossed like a stale donut at the back of the kitchen. Unfortunate, but sometimes a reality in conservation. Coming to founder effects, imagine a fun road trip where only the first few friends get to sing their favorite tunes. They pick the songs that shape the whole playlist for future journeys. But that means leaving out some fresh hits. Just a few individuals starting a new population can drastically change the genetic mix, leaving behind their sweet, catchy diversity behind. This is crucial for conservationists fighting for small or fragmented populations that often struggle with dwindling genetic variability. Inbreeding takes us to a family reunion where everyone's a little too close. The risks of harmful recessive traits begin to rise when relatives take a chance on love. It's like rolling the dice and coming up snake eyes a few too many times. When families intermarry, their offspring may inherit double doses of these recessive alleles, leading to genetic disorders. Think of it as a game of genetic roulette, but this time the stakes are far too high, resulting in inbreeding depression, a serious, sad outcome for many species. Historically, royals weren't exempt from the drama. The Habsburgs, a European dynasty renowned for their intermarriage, suffered from a nasty run of genetic disorders, definitely not the royal bloodline they expected. The plight of isolated island populations echoes this fate, showcasing elevated rates of genetic conditions. Societies worldwide have long known the dangers of inbreeding, likely observing the fallout before the science of genetics even existed. In modern times, genetic counseling provides cautionary tales and a firm thanks, but no thanks when it comes to cousinly romance. Diversity is like the secret sauce for species survival, acting as the safety net in the big circus of life. When our environment shifts, be it climate change or new ailments, a genetically diverse population includes those with the traits necessary to thrive. The survivors thrive, and their genes carry on, adapting over generations. In stark contrast, a similar population with low diversity faces existential threats like a domino effect. One virus could wipe them all out, leading to certain extinction, a dismal finale for any species. In the realm of conservation biology, experts hustle to preserve this genetic diversity, conducting breeding programs that play matchmaker for different individuals while quashing inbreeding. Some even bring in outsiders to reinvigorate the gene pool. Humans, amusingly enough, are not the most genetically diverse species, thanks to our rocky evolutionary ride. Yet this limited variation is a treasure trove of adaptations. Protecting our genetic diversity is like guarding a museum full of evolution's best artifacts, showcasing our rich history of survival against all odds. Every genetic bottleneck can be a wild card, substantially reducing diversity. Think of it as a drama-filled reality show where a sudden disaster eliminates half the cast, leaving only those lucky few who snagged a golden ticket. The survivors carry fragments of what was once a thriving genetic mix. Whether due to environmental disasters or isolation, each bottleneck shapes future populations. The drama and unpredictability of nature remind us that preserving genetic diversity isn't just a scientific endeavor, it's a race to ensure life continues in all its splendid performances. The drama of population genetics. When species bounce back from the brink, they often emerge from a narrow gene pool. Take cheetahs, for instance. They're living proof of an ancient genetic bottleneck. With all those spots resembling a bag of marshmallows, it turns out they're quite similar genetically, indicating that at some juncture, they got whittled down to a handful like the last chips in a snack bowl. The not-so-hilarious twist? This limited variety leads to a lot more hiccups in breeding and makes them susceptible to diseases. Then there's the northern elephant seal saga. It's a real rags to riches story. These magnificent creatures were hunted almost to oblivion in the 1800s, with the population sinking to a meager 20 individuals. Fast forward to today, and they've made a comeback to over 100,000. However, despite their triumph, genetic studies reveal they carry a hefty dose of genetic similarity from that tiny pool of survivors. They might be flourishing numerically, but their genetic diversity resembles a stale crouton. Of course, we humans are not off the hook either. In our compelling tale of survival, some believe our population once nosedived to just a few thousand. Forget the drama of gym memberships or fad diets. We've got a genetic signature that harks back to our near extinction. This lack of diversity is a reality check, reminding us we're not as unique as we like to think compared to other species strutting their genetic stuff on this planet. Speaking of strutting, let's chat about island populations. When a handful of adventurous souls decide to colonize an island, they bring with them a smorgasbord of genetic options. Except, it's more like bringing home leftovers instead of a full feast. And thanks to the founder effect, if one person brought a rare allele to the party, it might become the island's main dish, rare elsewhere but popular all around those sandy shores. Over the ages, these little island populations develop their own flair, setting themselves apart from their cousins on the mainland. Now, the Amish community provides a quirky illustration of founder effects in action. Descended from a small European immigrant group, they're like the exclusive club on genetic disorders, particularly when it comes to certain rare ailments that pop up more frequently among them. It's almost as if they inherited a quirky genetic lottery thanks to their founders bringing those particular alleles along for the ride. Migration, now that's where the comedy of genetics gets even richer. Every time someone moved across borders because of trade, the infamous wanderlust, or fleeing from melodramatic predicaments, new alleles were introduced, upping genetic diversity. These migrations have been our secret superpower all along, ensuring we remain one mishmash of a species. No matter the landscapes separating us, we can still mingle and spawn fertile offspring like a happy blended family. Today, migration continues to spice up our genetic stew. With people forging bonds across traditional barriers, we're experiencing a vibrant DNA jamboree. This mingling amplifies the celebration of our shared ancestry, proving at a genetic level, we're all just one big, merry clan. And in conservation efforts, keeping that gene flow alive is like throwing a lifeline to animal populations, connecting fragmented habitats and ensuring they don't drift too far into the genetic pool of doom. Finally, we must give a nod to natural selection, the true architect of survival stories. Beneficial alleles become the VIPs of the genetic game, taking center stage over generations. If they help organisms dodge predators, rustle up food, or woo their mates, then those lucky carriers. They're the ones who get the chance to pass on their success. Turns out, nature knows how to pick its winners, often with the finesse of a charismatic speed dater. Over time, the mighty allele spreads like wildfire through the gene pool, thanks to the forces of natural selection. Now, selection isn't just some boring science term. It's like the ultimate game show where the strongest contestants win. A spunky mutation that makes an enzyme more efficient or gives a protein a fancy makeover is suddenly a hot favorite. Even a teensy-weensy advantage can skyrocket an allele from rare to the hottest trend in town. Talk about genetic glow-up. But hold your horses. Not all selection is a joyful celebration. There's a dark side known as negative selection. Think of it as the bouncer at the club, kicking out harmful alleles so they can't crash the party. Most new mutations are just trying to fit in, but many are neutral or slightly harmful, so selection swoops in to weed them out, helping to keep those genes functioning smoothly. Like a dedicated editor, it ensures only the best make the cut and prevents the chaos of damaging changes from piling up. Then we have balancing selection, the true hero of genetic diversity. Economists would call this a win-win situation, where having a mix of different alleles in a population is advantageous. Take the famous sickle cell example. People with one normal and one sickle cell allele get sweet protection against malaria without suffering the severe symptoms of sickle cell disease. In regions where malaria's classic dance is dominant, both alleles happily coexist like invited guests at a cultural potluck. Heterozygote advantage is a superstar in the balancing selection realm. When having two different alleles gives you a boost compared to having two of the same, it's like the ultimate genetic Macarena. Yes, the sickle cell trait is the celebrity here, but it's not alone. Certain MHC genes, crucial for immune function, also strut their stuff with heterozygote advantage, allowing our immune systems to recognize a wider array of pathogens. In populations where this advantage thrives, even the bad alleles stick around, proving that fitness isn't so black and white after all. Genetic load, the baggage we all carry. It's like the quirky friend who comes along for the ride, no matter how awkward. Every person is walking around with some harmful mutations, adding to the genetic load of the population. Most are sneaky and recessive, causing no issues unless they find their matched twinsies. Others might throw a little tantrum with mild effects that don't completely ruin the party. But hey, mutations are relentless, popping up faster than you can say natural selection, creating a cocktail of genetic baggage we just can't shake. In small populations, this genetic load can become concentrated, which can be trouble like a bad case of overstuffed luggage. Suddenly, harmful alleles are more likely to show up in homozygous form, leading to health hiccups. This is why inbreeding can be a real buzzkill. It increases the chances of matching those pesky recessive alleles, making hidden issues rear their ugly heads. Time to call in outbreeding. Mixing with unrelated individuals is like giving diversity a high five, masking the genetic load and keeping it under control. Understanding genetic load has real implications, especially in medicine and public health. It suggests that each of us is a treasure chest of risk alleles, not just those with family dramas starring genetic diseases. As we delve deeper into genomic studies, we get a clearer picture of human health, unlocking mysteries and unveiling risks. Genetic counselors are like those wise friends who help you navigate this maze, assessing inherited conditions, explaining all the genetic jargon, and supporting your decisions like a trusty sidekick. When genetic testing is on the table, a counselor isn't just handing out numbers and graphs. They dive into a treasure trove of information. They break down what the tests can reveal and what they can't, helping families understand the maze of results that can come back. A positive result might send you into high alert mode, but it's not a sentence. Meanwhile, a negative might feel like hitting the lottery, but it doesn't mean you can throw caution to the wind. Risk always lurks in the shadows. Picture this, the anticipation of a baby's arrival, but first, let's talk prenatal genetic testing. With the magic of modern genetics, expecting parents can pinpoint potential issues early on. Tests are to the rescue, identifying chromosomal hiccups like Down syndrome or other genetic quirks like cystic fibrosis before that tiny bundle of joy makes their grand entrance. This empowers families to prepare and strategize, or at least stockpile baby blankets. Now, not all tests come wrapped in a gentle bow. Non-invasive prenatal testing sneaks a peek at fetal DNA in mom's blood. It's like getting a sneak preview of a movie. It's safe and straightforward, but some families brave the more invasive amniocentesis or chorionic villus sampling for a deep dive discussion. Sure, these methods carry tiny risks, but they offer a buffet of genetic info to chew on if you're ready for the feast. Of course, prenatal testing isn't just a game of numbers. It's a gut-wrenching decision showdown. With this data, the ethical questions rain down. What does one do with all this knowledge? Some folks may decide to continue their pregnancy, using the info to prep a superhero nursery, while others face harder choices. Each family has their own moral compass, and genetic counselors are there to help explore all paths with kindness and zero judgment. Fast forward a bit, and newborn screening takes the stage mere moments after birth. A quick blood test checks for a smorgasbord of genetic disorders, potentially nipping serious health problems in the bud. Think about PKU. Without early detection, phenylalanine runs wild, wreaking havoc on a baby's brain. But with proper screening and a specialized diet, the little ones grow up spouting nursery rhymes instead of neurodevelopmental woes. And don't forget, the screening kiddo keeps growing alongside our genetic knowledge. Now many regions can check for more than 60 disorders. It's a wonderful yet tricky landscape to navigate. The challenge? Balancing the exhilaration of early detection with the reality of false positives and the pesky stress that comes as an unwelcome guest to the party. Let's talk about a fairly modern twist on medicine. Pharmacogenomics, where our genes don't just shine light on potential health issues, but also play matchmaker for medications. Everyone reacts differently to drugs. A miracle for one could be a nightmare for someone else. Understanding these quirks means customizing prescriptions based on genetics, strategically heading off adverse reactions before they even kick in. Talk about personalized medicine. This is like crafting the perfect playlist for your body. At the heart of pharmacogenomics are the CYP450 genes, the unsung champions responsible for drug metabolism. These little guys can determine whether you're a speedy metabolizer, someone who needs more of that medicinal magic, or a slowpoke, requiring a gentler touch with lower doses or alternate meds. How cool is that? Tuning in to our genetic symphony makes all the difference in getting the best treatment without the unwanted drama. Pharmacogenomics is making waves in the worlds of psychiatry, oncology, and beyond. Imagine treating cancer with a customized approach based on the unique genetic mutations lurking in the tumor. And it doesn't stop there. When it comes to antidepressants, metabolic gene variants are calling the shots. As our grasp of genetics expands, medicine is increasingly striding toward personalized treatments that cater to the specific genetic fingerprints of patients. At its heart, cancer is a genetic rebel. It's really a party crash of mutations that disrupt the normal order. It happens when cells go rogue, disregarding the rules of controlled division and cell death. Generally, it's not just a solo act. Multiple mutations are needed for cells to throw caution to the wind. With each cell division, there's more room for errors, and over time, these accumulate, leading to the dreaded C word. And let's not forget about inherited mutations. Some folks are born with genetic red flags like the notorious BRCA1 and BRCA2 mutations, which put them at a heightened risk for breast and ovarian cancers. Or take a peek at Lynch syndrome, where DNA repair genes have a bad case of sloppiness, raising the stakes for colon and other cancers. Having these mutations is like carrying a loaded gun, not an absolute death sentence, but definitely ratchets up the risk factor. External culprits join the cancer party too. Say hello to tobacco smoke, UV rays, and the odd shady virus, all of which can cause DNA damage. It's a wild mix of genetic luck and environmental mischief, creating the perfect storm for cancer to thrive. Tumor DNA becomes the detective, revealing which mutations are calling the shots in this chaotic saga. When we sequence the tumor's DNA, it's like reading its diary, discovering the secrets behind its wild behavior and potential treatment paths. Not all mutations are born equal. Some are like the lead singers of a bad band, activating those pesky oncogenes that accelerate cell growth. Others are the drumbeats of destruction, silencing tumor suppressor genes intended to keep cell growth in check. Each type of cancer carries its own mutational signature, like fingerprints that tell tales of how the tumor came to be. For instance, lung cancers from smokers often bear the brunt of tobacco-induced mutations, while UV exposure gives skin cancers their own unique flair. The magic of precision oncology swoops in, wielding genetic info like a superhero cape. By matching therapies to specific mutations in a patient's tumor, doctors can crank up treatment efficacy while minimizing the potential for side effects from mismatched therapies. Targeted drugs battle particular mutations like a well-planned heist, especially in lung cancers with EGFR gene mutations, leading to better response rates for those lucky enough to carry them. Diving deeper, population genetics broadens the view. It's not just about individual mutations, but also about how gene frequencies dance within populations over time. It's a blend of genetics and evolutionary fun, delving into the myriad factors like natural selection, genetic drift, migration, and mutation. Populations buzzing with diversity are like treasure troves of genetic variations, enhancing adaptability. In contrast, low diversity feels more like a vulnerability waiting to unravel, leaving them at the mercy of diseases or other environmental upheavals. Population genetics is like the superhero of conservation, swooping in to save endangered species by ensuring their genetic diversity hangs on by a thread. Picture a team of quirky conservationists donning lab coats, armed with data and pizzazz, figuring out how to keep these precious creatures from going the way of the dodo. Thanks to population genetics, they're not just crunching numbers, they're crafting strategies that might just save the day. Then there's the Hardy-Weinberg principle, the star of the population genetics show. Imagine it as the rule book for how allele frequencies should ideally play nice in a population. It claims that if everything's perfect, no mutations, random dating, zero natural selection, and all that jazz, allele frequencies stay constant. Of course, real life is messier than that Sunday morning hangover, and populations rarely meet these clean-cut criteria. But when they don't, it's a neon sign that evolution is at work. When populations drift from the Hardy-Weinberg expectations, it's like nature's way of throwing a wild party. Maybe one allele is getting more love and attention because it's cute or fashionable. Perhaps there's some genetic mutation crashing the bash with new variants. Scientists can measure just how far a population has strayed from equilibrium, unraveling those sneaky forces that are causing the genetic ruckus. They can even use it to figure out how many people in the crowd are carrying recessive genes for genetic disorders, a neat trick for both genetic counseling and keeping public health in check. Speaking of tricks, get ready for genetic drift. Think of it as nature's dice game. In small populations, allele frequencies can change faster than you can say survival of the fittest, and that's purely by chance. Two alleles start off with equal flair, but through sheer luck, one allele gets all the spotlight while its competitor fades into obscurity. It's a wild, unpredictable world where fortune favors the randomly lucky. But wait, there's more. Picture a devastating bottleneck where a population shrinks to a handful of individuals. It's like nature's version of a reality TV show elimination round, leaving behind only a selected few who now carry a limited set of genes. These lucky survivors are tasked with reviving their lineage, though they're now carrying a genetic lottery ticket that may lack diversity. Similarly, founder effects pop up when a small group decides to start a new community, again leading to less genetic variety. Large, interconnected populations are allies in this fight for diversity and resilience. When families start playing the dating game too close to home, the dreaded inbreeding emerges, bringing along a cavalcade of problems. If two relatives mate, there's a higher chance their offspring inherit two copies of the same gene, including the not-so-great recessive alleles. This tricky situation can lead to inbreeding depression, showcasing a slew of genetic disorders and overall unhealthiness. Isolated communities and royal families have danced with this genetic dilemma for generations. Case in point, the infamous Habsburg dynasty, whose intermarriage led to noticeable genetic disorders. Their royal lineage, while glamorous, became a museum of health problems thanks to inbreeding. Likewise, island populations often find themselves in similar genetic bind, revealing that while keeping it in the family can be romantic, it's often a recipe for genetic chaos. Close inbreeding has been a universal taboo across cultures, and that's not just for dramatic family reunions gone awry. Long before the scientific community made sense of genetics, many cultures observed the not-so-awesome effects of closely related offspring. Nowadays, genetic counselors are the friendly neighborhood watchdogs, advising against close relative reproduction to sidestep any potential offspring pitfalls. And you know what they say, diversity is the spice of life, especially in the gene pool. Why does genetic diversity matter? Well, imagine a buffet where everyone is serving the same bland dish. No one's sticking around for seconds. When the environment throws a curveball, be it climate change, newfangled illnesses, or whatever else Mother Nature dishes out. Rich genetic diversity means some lucky individuals will have the moves to shimmy through the shakeup. Those adaptable souls pass on their genes, and voila, the population can keep on adapting. Meanwhile, a genetically homogeneous crew risks total wipeout. Any trouble will find everyone jumping ship at once. Now let's talk turkey about conservation biologists, the unsung heroes of genetics. These folks work tirelessly to boost genetic diversity, especially for endangered species. Their strategies involve all the good stuff: running breeding programs to keep inbreeding at bay, connecting fragmented critter populations for a little genetic intermingling, and sometimes even inviting new blood from other regions to the party. Think of them as the matchmakers of the animal kingdom. Surprisingly, humans aren't the genetic all-stars of the species world. Our diversity is pretty low compared to other critters. This sad state of affairs stems from a rocky evolutionary history and those pesky population bottlenecks. Our genetic patchwork is precious, however, representing how we've adapted to all sorts of environments. Maintaining our genetic diversity is like hanging on to a treasure trove of evolutionary tale stories that reflect our common history and global journeys. Speaking of genetic bottlenecks, they can occur when a population shrinks dramatically due to disasters or diseases, drastically cutting down the gene pool. Just look at cheetahs, who have remarkably similar genes and face reproductive and disease vulnerabilities due to their historic brush with near extinction. Similarly, northern elephant seals were nearly wiped out, dwindling down to just 20 individuals in the 1800s. Their comeback is impressive, but their genetic diversity today tells a sobering tale of survival from that tiny group. Humans, too, experience bottlenecks in our evolutionary journey. Some believe we might have plunged down to a few thousand individuals at one point. That genetic echo of a near extinction is what contributes to our current low diversity compared to all the other beings frolicking around us. It's kind of like wearing a vintage t-shirt, a stylish nod to our complex history. Let's not forget the intriguing dynamics of island populations, the ultimate examples of founder effects in action. When a small group of folks hitches a ride on a boat to a new locale, they bring along only a chaotic slice of the genetic pie. If one brave soul happens to have a rare allele, it can become the island's hottest trend. Take the Amish, for example. They're the proud heirs to a very select group of founders from Europe, resulting in a few unique genetic disorders popping up much more often than in the general population. Talk about a genetic history lesson. As humans journeyed across the globe, they carried with them a rich palette of genetic traits, shaped by the founder effects that we observe today. Each time groups broke off and established new populations, they brought along only a slice of the genetic pie. But never fear, migration swoops in like a superhero, mixing it all up again. When wanderers set up camp in new places and start families, they sprinkle in fresh alleles, enhancing diversity and knitting tighter connections among us all. Migration isn't just an adventure. It's like nature's way of throwing a melty genetics party. As history unfolds, people have been trekking for trade, conquest, exploration, and sometimes just to escape a wacky situation. Whether they've been traveling on foot, by ship, or via the latest high-tech transportation, these movements have continuously drawn populations closer, preventing the rise of separate human species. Today, thanks to ongoing migrations, we're still a colorful patchwork of one interconnected global family. Moving on, imagine gene flow as a party crasher at the genetic gala. It keeps the dance floor alive by ensuring populations don't become awkward wallflowers. Migrating individuals add a delightful array of genetic flavors, preventing any monotony in the gene pool. Nature truly loves a good reunion, as these shared genes carry along beneficial traits that can help populations adapt over time. Just like mingling at a party, this genetic mixing enriches our heritage in the ultimate social or genetic gathering. In the wild world of conservation, gene flow plays a vital role too. Picture animals trying to mingle in fragmented habitats like individuals at a chaotic wedding reception. Corridors designed for animal movement ensure they don't get stuck in isolated corners, avoiding the perils of inbreeding and genetic dullness. These lifeline pathways sustain genetic vitality and diversity across landscapes, keeping populations' dance cards full and vibrant. Now let's chat about natural selection, the ultimate filter at the genetic dance-off. It favors alleles that make organisms more adept at dodging predators, securing meals, or wooing mates. Over generations, those savvy alleles get the spotlight and become more prevalent in the population. A little evolutionary boost can send a previously rare allele skyrocketing into the limelight. But watch out, negative selection is lurking around, ready to kick harmful alleles to the curb, ensuring that only the best genetic contenders take center stage. Diversity in genetics sometimes works like an intricate balancing act. Take the sickle cell trait, for example. It showcases how certain alleles stick around because of heterozygote advantage. Individuals with one normal and one sickle cell allele enjoy a protective layer against malaria without suffering from severe disease symptoms. This balancing act keeps a mix of alleles thriving, ensuring that populations retain diversity like a well-stocked buffet where everyone finds something to enjoy. Heterozygote advantage proves that genetics can be cheeky, reminding us that context matters. An allele that shines brightly in one setting may fall flat in another. Dilemmas arise in disease genetics, as alleles that seem troublesome might hang around for their secret perks. Appreciating these genetic nuances is crucial for predicting health risks and guiding genetic counseling. In summary, evolution is a whimsical game, ensuring that while there's no one-size-fits-all allele, each one strikes a harmonious chord in its unique context. Genetic load is like that unwanted baggage we all carry around without even realizing it. Every one of us is a walking collection of DNA quirks, featuring various mutations that might not cause any drama until paired up with another pesky mutation lurking in our genes. Most mutations are sneaky recessive types, quietly hanging out until they decide to throw a party, resulting in potentially significant health issues when two copies get together. Imagine this: mutations pop up faster than you can say oops, while natural selection applies a gentle, yet imperfect hand. New mutations are just waiting to cause trouble, especially since many are rare or have minor side effects. In larger gene pools, these mutations spread out like a burrito filling, but in smaller populations, they tend to huddle together, creating a higher risk for harmful traits to mingle and manifest. This is where inbreeding takes a spotlight, dramatically increasing the chances that these troublesome alleles join forces and turn into real problems. Outbreeding is like hitting the genetic refresh button. Engaging with unrelated individuals gives those pesky deleterious genetic alleles less opportunity to become partners in crime. Think of it as a family reunion where the more diverse the crowd, the less likely the awkward conversations are going to arise. This genetic mingling disguises the bulk of genetic load, and suddenly our genetic genealogy becomes a colorful tapestry of health outcomes rather than a gray blanket of inherited glitches. When it comes to our health and the medical world, genetic load has a whole lot of implications. It's not only the folks with known family histories of diseases who carry the risk alleles. Surprise! Everyone could be carting around some genetic surprises that would make a soap opera addict blush. The realm of genomic studies is bursting with information, which helps paint a more panoramically accurate picture of human health and the myriad risks that tag along. For those diving into the deep end of genetic waters, genetic counselors are like your savvy lifeguards. They specialize in helping families decipher this intricate genetic lingo, assessing risk, explaining test results, and guiding individuals through the turbulent waters of genetic decision-making. Starting with a detailed family history session, these pros construct family trees that reveal who in the clan has dealt with various health conditions, acting like a roadmap to the inheritance game. Once testing is on the table, genetic counselors break everything down into bite-sized pieces. They clarify what a test can actually show, the different results that might roll in, and what the findings could mean for the individual and their family. It can be a wild ride, and they're there to help families coast through the emotional roller coaster of learning about their genetic risks, offering support while respecting personal choices. In recent years, prenatal genetic testing has emerged like a superhero in a lab coat, revealing whether a fetus might be grappling with specific genetic conditions. Non-invasive tests are particularly popular as they gather fetal DNA floating around in the mother's bloodstream, allowing for early screenings without the drama of more invasive methods like amniocentesis, which come with a few hang-ups of their own. These advancements create new ethical dilemmas. as families grapple with the kinds of decisions they might have to face based on test results. With all this talk of spotting genetic conditions before birth, we enter a labyrinth of choices. Each family's values can lead to a different journey, whether that means preparing for a child with a condition or navigating difficult decisions about pregnancy continuity. As testing capabilities expand and start peeking at predispositions for late-onset diseases or mild nuances, the debate continues: how much information is too much, and how do families find clarity in the jumble without feeling overwhelmed? Such is the fascinating world of genetics, where even the tiniest details matter. Shortly after making their grand entrance into the world, newborns undergo a super important test called newborn screening. This blood test checks for dozens, yes dozens, of genetic and metabolic disorders. Imagine a tiny needle prick to their heel leading to a treasure trove of health data. For conditions like PKU, where babies can't break down an amino acid called phenylalanine, early detection is key. Left unchecked, the amino acid could wreak havoc, but with a special diet, little ones can flourish like wildflowers. Among the other conditions lurking in the shadows are sickle cell disease, congenital hypothyroidism, and cystic fibrosis. Thanks to the magic of early detection and timely treatments, countless lives have been transformed. It's like we've hit the health jackpot, protecting millions of children and ensuring they get the best possible start in life. As scientists uncover more genetic mysteries, the list of conditions tested keeps expanding, with some regions checking for over 60 different disorders talk about health goals. But let's not forget the flip side of this coin: false positives. No one wants to worry needlessly or face the financial burden that comes with expanded screenings. Like an overzealous alarm system blaring at every small thing, it's all about finding that sweet spot between beneficial early detection and the horrors of unnecessary panic. Still, the promise of preventing serious health issues is a worthy goal, because nobody wants to be in the position of saying, "Oops, we jumped the gun." Enter pharmacogenomics, the cool science of tailoring medications to your genes. Have you ever noticed how a drug that works wonders for one person might flop for another? That's genetics talking. Pharmacogenomics digs into how our genes influence drug metabolism and efficacy, ensuring we get the best treatment tailored just for us. Imagine doctors peering into a crystal ball formed from your DNA to predict the right medication dose, reducing adverse reactions like the morning after hangover no one asked for. A prime example of this is the CYP450 gene family, responsible for metabolizing a plethora of medications. Variants here can make someone a speedy metabolizer, chomping down medications like candy, or a slow one, treating drugs like they're fine wine better enjoyed in moderation. This personalized approach is spreading like wildfire through psychiatry, oncology, and other medical realms, because everyone loves a good tailored suit, and healthcare should be no different. But here's the plot twist: cancer is a genetic chameleon. It's all about accumulated genetic mutations that turn our cells into unruly party crashers. These mutations typically impact genes that control cell division, DNA repair, and cell death. Cells come equipped with multiple safeguards, hence why it often takes a series of errors before cancer struts onto the stage. It's almost like they're a rock band, lots of practice needed before they can hit the high notes of cancer dominance. And here comes the age kicker: the risk of cancer increases as we pile on the birthdays. With each cell division, there's a chance for mutations, and over a lifetime, they tend to accumulate like laundry. You might also inherit some predisposing mutations like BRCA1 and BRCA2, which ramp up breast and ovarian cancer risks. Add in environmental factors like tobacco, UV rays, and pesky viruses, and you've got a real genetic roller coaster, one that merges the realms of nature and nurture to create the ultimate battle of DNA. When a tumor pops up, it doesn't just crash the party uninvited. It brings its own set of DNA mutations. By sequencing this rebellious tumor DNA, scientists can pinpoint which genes are acting out. This playful game of find the culprit helps reveal how the cancer got into trouble and suggests ways to put it back in line. Some mutations get those pesky growth-promoting genes fired up, while others silence the growth-stopping genes. Talk about a wild party. But hold on to your lab coats, because not all cancers are created equal. Each type has its own signature mutations, like unique fingerprints that tell the tale of how it misbehaved. For instance, lung cancers from smoking have distinct mutations linked to those smoky party crashers. Meanwhile, skin cancers chilling in unexposed areas have a different story to tell thanks to those sunbathing UV rays. These mutation signatures are crucial breadcrumbs on the path to understanding and treating the tumor. Now, let's talk about targeted therapies, the superhero of cancer treatment. By decoding the genetic makeup of a tumor, doctors can craft a precisely targeted attack plan. Some drugs are the equivalent of heat-seeking missiles for cancer cells. Take EGFR inhibitors, for example, perfect for lung cancers packing the EGFR mutation. Patients lucky enough to have this mutation can respond like champs, while those without it might as well be throwing spaghetti at the wall. It's precision oncology in action, making treatments personal. Switching gears to the crime scene, forensic genetic steps in like a detective with a magnifying glass. DNA is the ultimate unique identifier, working wonders by linking clues found at crime scenes to potential suspects. Imagine finding a tiny DNA trail on a weapon or a shirt bam. Enter DNA profiling, the high-tech method that constructs a genetic fingerprint from those specific regions of the genome, nabbing criminals and releasing innocent folks from behind bars. But even with its superhero status, using DNA in law enforcement stirs up some moral dilemmas. Contamination, errors, and shaky statistics can plague evidence, while the use of genetic databases often treads a fine line between safety and privacy. Officers may solve cold cases with creative genetic genealogy, but implicating innocent family members raises eyebrows. Justice must balance on one side, while personal rights hover precariously on the other, quite the juggling act. Paternity testing is a tale of DNA determining who belongs where. When a child's paternity is in question, DNA testing swoops in to compare genetic markers between child and father. These tests are like the referee in a family dispute, with match probabilities soaring over 99.9%. But that's just the tip of the iceberg. Siblings can be revealed, and hidden family ties uncovered, leading to heartwarming yet sometimes emotionally complicated revelations. And let's not forget about ancestry testing, the ultimate ancestral treasure hunt. By analyzing your own genome, these tests unveil your ethnic background and connect you with long-lost relatives. Picture comparing your DNA to a plethora of reference populations worldwide. It's like embarking on a global safari of family history. But beware, the thrill of discovery can sometimes unearth secrets, sparking everything from joyous reunions to jaw-dropping surprises that reshuffle family dynamics like a game of cards. Imagine diving into a treasure chest of your family history, where you discover that your great-great-great-grandma isn't just some mystery lady in an old photo but a vibrant part of a global tapestry. Ancestry testing peeks into your genetic past, offering a playful peek at where you and your genes might have traveled through time and space. Companies like 23andMe or Ancestry.com have cracked the code using genetic variation patterns, allowing them to identify the regions that may shape up your heritage. The more data they accumulate, the sharper their crystal balls become. Ever thought you were related to royalty? Well, thanks to these genetic tests, many people have reconnected with long-lost relatives or stumbled upon previously unknown branches of their family trees. It's like opening up an unexpected gift basket filled with second cousins and quirky family tales. By matching your DNA with others in a huge database, they can pinpoint people who share similar chunks of DNA, leading to those delightful holy guacamole, you're my cousin moments. Sure, these ancestry estimates aren't foolproof. They're more like friendly guesses than hard facts, but who doesn't love a good family reunion? Now let's dive deeper literally. Mitochondrial DNA, passed down from mother to child without tinkering around, is like a time capsule that connects us to our maternal lineage. Imagine tracing that line back thousands of years and discovering that your ancestors might have wandered from Africa, journeying through the Middle East, or even crossing into Europe during the Ice Age. Sounds like a captivating sequel to your family saga, right? These stories of deep ancestry can be cool, collective narratives that give you a sense of connection to a broader human heritage. And for the gents out there, Y chromosome analysis has got your back, and your lineage. The Y chromosome is handed down from father to son, preserving family ties through generations. By identifying specific mutations, you can trace where all those male ancestors might have strutted their stuff over the centuries. It's like a genetic GPS that not only reveals your paternal lineage, but also provides a front-row seat to historical events like how agriculture or empire expansions influenced your ancestors. Speaking of events, there's a fascinating tale involving Genghis Khan. Did you know that his Y chromosome lineage is a genetic rock star in Central and East Asia? Studies suggest this lineage spread from a single ancestor about 1,000 years ago, potentially tying back to Khan himself. While we can't exactly roll back the years for confirmation, the clues paint an intriguing picture. So next time you see your strong chin in the mirror, remember, it might just come with a dash of Mongolian flair. With our ancestry tools, we also encounter a humbling truth: everyone's male ancestry can be traced back to a common Y chromosomal atom. So, as you sashay through family histories, remember, ladies, there's a patriarchal node in your vast family tree, weaving together all the living men. It's a whimsical notion that every male is simply a branch in the intricate family tapestry stretching back eons. Now, about that DNA, it's somewhat of a diva. While it can survive for millennia in the right conditions, heat and moisture can make it vanish quicker than your willpower in a bakery. So while the prospect of connecting with ancestral populations through DNA can be thrilling, it's crucial to note that in its fragile existence, DNA can be fickle. It might decay in just a few decades or centuries in less than ideal conditions. Nevertheless, as we unravel the genetics of our origins, we get a gripping glimpse into our shared human story. In the chilly embrace of permafrost, oxygen-poor caves, or the arid expanses of deserts, ancient DNA has found a remarkable sanctuary. While most of this DNA is somewhat younger, between thousands up to tens of thousands of years old, scientists have extracted DNA from mammoth teeth preserved in Siberian permafrost for over 1 million years. It's like the ultimate frozen treat for geneticists, offering a glimpse into an era long past, albeit in a somewhat jigsaw puzzle state. Unveiling the past through ancient DNA isn't merely an academic exercise. It's a wild adventure into human history. Bones and teeth uncovered from archaeological hotspots have offered juicy tidbits about our ancestors' genetic makeup, detailing migration patterns and the fascinating dance of social interactions between groups like Neanderthals and modern humans. What a cosmic soap opera it is, learning about how our ancestors mingled and jostled their way across the globe while rocking the half-primitive, half-modern fashion statements of their time. However, the challenges of extracting ancient DNA are no laughing matter. Imagine a real CSI, ancient history. Modern DNA contamination lurks around every corner, and since ancient samples tend to be fragmented like last week's jigsaw puzzle, scientists must wield specialized techniques to recover the remnants. But fear not. Thanks to cutting-edge tech, they've turned retrieving these precious genetic jewels into a formidable quest, unearthing clashing stories of ancient lives. Among the greatest plot twists is the revelation that modern folks today carry a smidge of Neanderthal DNA, about 1% to 2% of our genetic makeup. In fact, after modern humans embarked on their great African exit, they crossed paths with Neanderthals, especially where the Middle East is now. Talk about a surprising family reunion. Some Neanderthal genes turned out to be vital players in improving immunities, which shows how human history isn't just one long line. It resembles a complex web of genetic interactions. In addition to the Neanderthals, ancient humans like the Denisovans also contributed their DNA to the mix. Fast forward to modern-day Southeast Asians, Oceanians, and Australians. Surprise. Their genes indicate a rich history of interaction with the Denisovans. It's wild to think that certain Denisovan genes are linked to high-altitude adaptations in Tibetans, highlighting how interconnected we all are through shared ancestry and genetic loves of yesteryear. Then we have the domestic drama of our four-legged pals. Genetic studies on domestication unveil how wild creatures morphed into the companionable pets and livestock we adore today. Dogs, our furry best friends, were the first to cross over from wild to domesticated, dating back over 15,000 years ago. It's fascinating to see how selection influenced behaviors. Those floppy ears and unique coat colors weren't just happenstance. They were like nature's very own makeover show. Yet, it's not just pups getting genetic makeovers. Every domesticated animal, cats, pigs, cattle, and chickens has a unique tale in their genes, some having been domesticated multiple times across the globe. By examining the genomes of these critters, scientists have uncovered the traits that were specifically selected during domestication, painting a picture of our intertwined fates. The lineage from wild ancestors to domesticated friends speaks volumes about our evolving relationship with animals. Don't think the plant kingdom gets left out of the fun. Domestication has dramatically reshaped the crops we rely on, transforming wild grasses into delicious staples. Take corn, for instance. Once just a wild grass called teosinte with tiny, hard seeds, today's corn has bloomed into juicy cobs filled with accessible kernels. It's truly a testament to humanity's tremendous power to shape the natural world through keen observation and selective cultivation over thousands of years. Genetic studies make it quite clear. A select few genes have gone through the ultimate makeover during the domestication of plants like corn and wheat, a process akin to turning a wild party into a sophisticated soiree. These genes were targeted for significant effects, playing a role in everything from seed size to toxicity. Wild plants often come equipped with defenses like thorns and bitterness, making them about as palatable as a cactus salad. Domesticated varieties, however, ditch the defense mechanisms in favor of edibility, making them prime candidates for human consumption. Here's a fun twist. Some of these domesticated plants have become so snugly reliant on humans that they can't even handle their own reproduction without our good old assistance. Take maize, for example. It needs a helping hand to shed those seeds like a child needing a nudge to share their toys. And then there are bananas, which, lacking pesky seeds, have turned into a smooth, seedless pleasure that prefers vegetative propagation. These plants really are the ultimate indoor pets, genetically crafted to thrive alongside humans, a partnership as old as time itself. Now, let's shift gears from crops to critters. Genes don't just influence our vegetables, they have a say in behavior too. Genetic components play a role in how all living beings present themselves. Research in both animals and humans showcases that genes can shape temperament, learning cravings, and even social dynamics. It's a wild cocktail of genetics, environment, culture, and experience, all swirling together to create our behaviors. In lab mice, specific genetic mutations can lead to increased anxiety or aggressive tendencies. Talk about a nervous little rodent. In humans, the family tree of behavioral genetics unfolds delightfully, with twin studies revealing the nuances of genetic influence on various traits. By taking a peek into the lives of identical and fraternal twins, researchers can decipher just how much of our individuality comes from our genes. They suggest that even complex traits like intelligence and personality have their roots in genetics. But, and this is a big but, interpreting this bounty of data requires a healthy sprinkle of caution. Heritability is context-specific. It reflects variations within populations, but doesn't seal the deal on traits being unchangeable. Speaking of intelligence, the genetic machinations behind this elusive trait have sparked endless debates. Yes, studies show that intelligence, however you define it, has heritability. It's rooming with a host of genetic variants. But each of these variants contributes merely a drop in the ocean, illustrating the mind-boggling complexity of what makes us us. Adding to the fun, environmental factors like education and nutrition jazz things up even more. It's a big party where the genes do their dance alongside influences from our surroundings. However, diving into discussions of group differences in intelligence can lead us down a slippery slope. While some may shout from the rooftops claiming genetic differences between populations, science shows us that these assertions lack substantial backing and tread into ethically murky waters. Intelligence is multidimensional and tricky to pin down. It often gets lost in translation through tests that may reflect cultural biases more than cognitive abilities. Besides, those minor genetic differences among populations stand small against the backdrop of a mighty environmental influence. The grand narrative of genes and environment unfolds in a beautifully intertwined dance. It turns out that nature versus nurture is less of a showdown and more of a celebration of cooperation. Genes and environment aren't adversaries. They play off each other all the time. How an individual reacts to their surroundings is influenced by their genes, just as the environment can impact how those genes express themselves. A classic example is phenylketonuria, an inherited condition where affected folks can't break down phenylalanine. This is just one snapshot of how genetic dependencies can interact with environmental factors, showing us the rich complexity of life. In the vast universe of genetics, the curious case of phenylalanine stands out like a beacon. Normally, when phenylalanine builds up in the body, it can develop into brain damage, a pretty grim outcome. However, toss in a low phenylalanine diet, and voila, a modified environment allows individuals to thrive. This magic trick demonstrates how the surroundings can influence genetic quirks, making certain traits appear only when the conditions are just right. But hold on, this isn't just a tale of phenylalanine. The story gets juicier when we look at other traits and their dance with the environment. The gene-environment interactions are endless. For instance, a variant that sparks skin cancer flares up in sunny locales, while a genetic tendency to pack on the pounds seems particularly zealous in the land of buffet lines and comfy couches. It's like the environment unleashes certain traits, making understanding these interactions crucial for healthcare and education. Epigenetics jumps in like an unsung hero, serving as the molecular bridge linking our genes with Mother Nature's mood. Picture this. Environmental factors like food, stress, and pesky toxins can tweak your epigenetic marks and influence how genes express themselves, all while the DNA remains unchanged. It's mind-boggling to think that those early life experiences, like a mom's nutrition during pregnancy, can set off lifelong impacts on our health and behaviors. Diving into the animal kingdom offers vivid illustrations of these concepts. For example, rat pups who bask in ample maternal attention develop unique epigenetic marks and stress responses compared to their neglected peers. The difference is environmental yet leaves a lasting imprint, much like a signature etched in the sand. The big question buzzing among researchers is whether humans experience a similar phenomenon to the same extent. The grand quest to decode our genetics took a giant leap with the Human Genome Project, a colossal collaboration that wrapped up in 2003. Think of this as the ultimate Yelp review for humanity's genetic makeup, identifying 3 billion base pairs and mapping all the genes in meticulous detail. Surprisingly, the genetic treasure map revealed that humans have far less genetic material than once thought, around 20,000 to 25,000 genes, which is barely more than a humble worm. The essence of our complexity lies not in the quantity of genes, but in how they regulate and interplay with one another. As the veil lifted from our genes post-Human Genome Project, the realization dawned that most of our DNA doesn't encode proteins at all. Once dismissed as junk, this non-coding DNA has emerged as a critical regulatory force, housing elements that influence gene expression like a maestro orchestrating a symphony. Unraveling this vast regulatory landscape has become the new frontier in genomics, a thrilling adventure just waiting to unfold. And here's where things get really exciting. Genomic medicine is the superhero reshaping healthcare as we know it. By delving into a patient's genome, doctors can craft tailor-made treatments, predict disease risks, and even diagnose rare conditions as if they're playing their own version of genetic Wheel of Fortune. In the cancer realm, tumor genome sequencing takes center stage, pinpointing mutations that can be targeted by specific drugs, revolutionizing treatment and boosting patient outcomes. The realm of rare diseases has been transformed by whole genome sequencing, which acts like a skilled detective exposing genetic causes behind multiple conditions. Once lost in the diagnostic labyrinth, patients can now receive accurate diagnoses straight from their genome, tying up those diagnostic odysseys with a neat little bow. This personalized approach to medicine isn't just a trend. It's becoming the new norm, crafting a brighter and healthier future for all of us, one gene at a time. Pharmacogenomics is like having a personalized chef for your medication. It tailors drug selection and dosing based on your unique genetic recipe. By understanding how your body metabolizes drugs, we can not only avoid nasty side effects but also enhance how well the medications work. With pharmacogenomic testing gaining traction, it's like your doctor is finally reading the fine print of your DNA and prescribing medicine that actually fits you like a glove. Imagine a future where healthcare feels less like one size fits all and more like a tailored suit. That's the vision driving genomic medicine, where treatments evolve based on individual genetic blueprints. We're not quite there yet, but swift advancements are paving the way. As the costs of genomic testing tumble and our understanding deepens, the dream of personalized medicine is about to become a reality faster than you can say caffeine-induced genetic variants. Way back when the Human Genome Project kicked off, sequencing DNA was the equivalent of waiting for a sloth to finish a marathon. Took years and cost billions. Fast forward to today, and guess what? Sequencing a whole genome can be done in a matter of hours for around $1,000. Thanks to new tech wizards and next-generation sequencing machines that can handle millions of DNA fragments at once. Translating your genetic code has never been easier or cheaper. It's practically a genome on a budget. This surge in affordability means we're now swimming in genomic data, enough to fill libraries. With genomic databases bursting at the seams, researchers have a treasure trove of genetic information to explore. Direct-to-consumer testing has put genetics in our hands literally. Clinical sequencing is becoming a staple for diagnosing illnesses and determining treatments. Now, imagine if we sequenced every newborn's genome, creating a health roadmap for life. But wait, what about data privacy, equity, and management? These are the million-dollar questions being tossed around the medical community. With all this data flying around, we've got a pressing issue. How do we make sense of it all? Enter bioinformatics, our data detective hero. Single human genomes produce a staggering amount of information, leading to gigantic projects that generate petabytes of data. Bioinformaticians whip up algorithms to align sequences, uncover variances, and even predict gene functions. They're the ones who create databases that are more organized than your closet, making sure this colossal amount of information becomes accessible and usable for scientists. The field is evolving, leveraging machine learning and artificial intelligence, which can spot patterns and make predictions that human eyes might miss. This computational prowess is becoming just as critical as bench lab work. The combination of biological research and computer science is like peanut butter and jelly, turning once impossible biological questions into manageable puzzles. As researchers struggle with an information overload, bioinformatics is emerging not just as important, but essential. Now, let's not forget about the unseen players in our health saga. Our tiny friends, microorganisms. Metagenomics is the fancy term for studying the trillions of bacteria, viruses, and fungi snuggled within our ecosystems, both inside our bodies and out. These tiny creatures, forming what we call the microbiome, have a huge impact on our health. From helping you digest your food to influencing your mood, these minuscule entities play major roles in our overall well-being. But the metagenomics mission doesn't stop at human bodies. Researchers are diving into environmental microbiomes, like those found in soil, oceans, and even the air we breathe. These little guys are heavy lifters in nutrient cycling, organic matter breakdown, and climate influence. By sequencing DNA from various samples and mapping out these microbial communities, metagenomics is opening doors to understanding how these microscopic teams contribute to health, environment, and everything in between. Who knew such tiny organisms had such a big role on the grand stage of life? The unseen world of metagenomics takes us on a wild ride into the vibrant realm of genetic diversity that underpins ecosystems. Imagine your microbiome as a lively sidekick, your very own second genome. While your human genome remains a set deal from the moment of conception, your microbiome is like a chameleon, adjusting itself through diet, antibiotics, and lifestyle changes as you navigate life. Essentially, you sculpt this microbial entourage, and they just might have a say in who you become, making things quite the partnership. Hold onto your hats because the numbers are mind-boggling. The gene count in your microbiome far surpasses your own, like a concert where the microbial stars steal the show. And what do those tiny musical maestros do? They craft enzymes and metabolic pathways your own cells can't handle. For example, gut bacteria are essentially fiber's best friends, breaking down complex carbohydrates to extract nutrients your body wouldn't otherwise digest. In other words, thank your microbes for those extra calories. The quest for health has now taken a compelling twist as researchers dive into manipulating the microbiome. Enter probiotics, those little heroes promoting a harmonious balance, and fecal microbiota transplants, which sound a bit like a horror movie but are indeed helping treat stubborn infections by transferring microbiomes from healthy donors. And guess what? Personalized diet plans based on your microbiome's unique composition are on the rise, making Know Thyself sound a tad more appetizing. But wait, this microbiome saga has a deeper layer. It's not just you versus your genes. We're talking about a friend group full of tiny partners that influence health and disease through an intricate dance of co-evolution. Your genetics vibe with microbial genes, forming a complex tapestry where bacteria transfer genes both vertically, parent to offspring, and horizontally, bacterium to bacterium, creating a whirlwind of genetic exchange. Who knew being social could get so microbish? Now let's get a bit spicy. Horizontal gene transfer is no small fry. It's a major player in bacterial evolution and antibiotic resistance. Picture this. A stubborn bacterium evolves resistance and then generously shares its resistance gene with others, even those from different species. Voila! You've got a speedway of resistance that speeds along faster than a gossip chain, posing a meaty challenge in our ongoing medical battles. And this isn't just a localized issue. It's a worldwide health crisis in the making. With some infections outsmarting our current antibiotics and new drugs always facing an evolutionary showdown, we've got ourselves a race against time. To outmaneuver these crafty bugs, we need to use antibiotics wisely, complete our prescribed courses, and focus on hygiene and vaccinations. Because in the grand scheme of things, evolution is relentless, always creating new twists in the tale. But let's not forget about the viral villains lurking around. While viruses aren't your average cells, they pack a punch with their own genomes, either DNA or RNA. These tiny genetic packages may be small, often just a few essential genes, but they come equipped with everything needed to commandeer a host cell. Having a knack for adaptation, viruses are quite original in turning hosts into gene-replicating factories, leaving us to marvel at their bizarre cunning. Who knew genes could create such a ruckus? When a mischievous virus breaks into a cell, it doesn't just waltz in. It injects its entire genome to take control. The viral genes are like demanding little divas who command the cell's machinery to whip up viral proteins and replicate themselves over and over. Before you know it, brand new virus particles are springing into action, busting out of their host and looking for fresh cells to infect. Talk about a rather catastrophic house party from the cell's perspective. Now, let's not forget how adaptable these pesky viruses are. RNA viruses, in particular, are notorious for their fast-paced mutating antics. Unlike cellular organisms, they lack efficient proofreading mechanisms, allowing mistakes to pile up like laundry on a Sunday morning. This rapid evolution enables them to skip past immune defenses, hop between hosts, and even laugh in the face of our drugs. That's why we must make a point to update our flu vaccinations every year. Those viral party crashers keep reinventing themselves. What's even wilder is when viruses decide to hit the ultimate hide-and-seek jackpot by integrating their genomes into the host's DNA. Retroviruses, such as HIV, cleverly copy their RNA genome into DNA and nestle it into the host chromosome like a secret agent. This sneaky strategy allows them to stay out of sight, quietly lurking until they're ready for a big comeback. To combat these viral tactics, understanding viral genetics is incredibly vital for crafting effective treatments and vaccines. In a twist of fate, some of these viral intruders have embedded themselves deeply into human history. Around 8% of our genome actually consists of sequences from ancient retroviruses. Known as endogenous retroviruses or ERVs, these are broken remnants of infections from our ancestors millions of years ago. If one of those pesky retroviruses made it into a sperm or egg cell, its DNA could be passed down, becoming a permanent coat of genetic armor in our DNA. Though most ERVs have malfunctioned over the years, their presence gives us a peek into prehistory. And hold on to your hats, because some of those ancient viral sequences have even been co-opted for beneficial functions. For instance, one ERV-derived gene is essential for placenta development in mammals, crafting the boundary between mother and fetus. Talk about viral genes finding new gigs. These ERVs are like old pets living comfortably in their owners' homes, reflecting our intertwined evolutionary stories. This sharing across species is how we trace back our molecular fossils, recording viral history like time travelers. Let's shift gears to RNA, the wild child of genetic material. While most living beings have settled down with DNA, certain viruses prefer the breezier, single-stranded RNA. RNA might be less stable than its DNA counterpart, but it's incredibly versatile. This rebellious molecule shows up in viruses like influenza, HIV, and coronaviruses, encoding proteins and replicating itself, sometimes even morphing into DNA in retroviruses. Its role in life's origin stories sparks endless fascination, hinting at a time when RNA ruled supreme. Moreover, RNA's capabilities aren't just about viruses. They play a vital part in every living organism's genetic symphony. Messenger RNA transports secrets from DNA to ribosomes, while transfer RNA helps translate those secrets into action. Then there's ribosomal RNA, the true star player in the ribosome's catalytic core. Even in the grand narrative where DNA claims most of the stage, RNA crafts a crucial role in the dance of life. Lastly, we can't overlook the magic of gene regulation, what makes every cell in a multicellular organism sing a different tune despite sharing the same genetic score. Thanks to fascinating mechanisms like chromatin structure and protein stability, gene regulation fuels cellular diversity. Transcription factors, the crafty proteins that bind to DNA, decide whether genes are turned on or off, sculpting a cell's unique identity. Isn't it wild to think that amidst this chaos, our cells find a way to harmonize their genetic orchestra? Muscle cells are the party animals of the cellular world, strutting around flaunting transcription factors that activate all the cool muscle genes. Neurons, on the other hand, are like the introverted guests at the same party. They express the factors that power up their own neuron genes. Enhancers and silencers, the regulatory DNA sequences, play the role of the event planners tweaking gene activity. These sequences might be hanging out miles away from their target genes, but magic happens as they loop through space to interact with promoters, ensuring that everything moves smoothly. The three-dimensional organization of our genome is nothing less than an intricate dance, critical for proper gene regulation. If there's a misstep in the choreography, it can lead to quite the disaster. Imagine a gene showing up at the wrong time or place. You might end up with a very confused development process. When tumor suppressor genes go into hiding or oncogenes throw a surprise party, cancers can crash the scene. Grasping how gene regulation works is essential for understanding how cells keep their identities while adapting to their environment. Now, let's throw in the transcription factors, the tiny maestros orchestrating developmental symphonies. They're like head conductors at a concert, coordinating the grand expression of a multitude of genes. During development, these factors start a cascade, with early birds turning on later ones, tining up waves of gene expression that sculpt the developing embryo. Just think about the amazing hawk genes, the rock stars of the gene world. They specify body segments, defining structures along the head-to-tail axis. If they go rogue, it could lead to critters with extra wings or limbs sprouting in the wrong places. Staying on the topic of transcription factors, we can't forget MyoD, a superstar that showcases the ease with which a single factor can completely transform a cell. It takes fibroblasts, those flexible connective tissue cells, and turns them into muscle cells like flipping a light switch. This ability to reprogram cell identities highlights the sheer power these regulatory proteins possess. By decoding networks of transcription factors, we can unlock secrets of complex organisms blossoming from just a single cell. It's a promising pathway toward regenerative medicine, where manipulated factors might help regrow damaged tissues or even sprout whole new organs. As if that weren't enough, let's turn up the dial on protein diversity with alternative splicing. Imagine a single gene being the ultimate remix artist, spinning out multiple proteins from one groove. Post-transcription, the RNA gets a makeover out with the non-coding regions, introns, and in with the coding exons. But here's the twist. Not all exons have to stay on the track. Cells often skip portions, mix and match exons, and each tune they produce transforms into a unique protein with its own flair and purpose. This nifty process is one of the reasons we have far more protein diversity than the rough estimate of 20,000 genes suggests. Beyond just remixing, alternative splicing keeps a tight fist on regulation, since errors in this process can wreak havoc, leading to genetic disorders. Fear not. Scientists are already exploring ways to manipulate splicing patterns, developing drugs aimed at correcting splicing issues. They're hoping to turn potential disasters into triumphs through the art of splicing. Now, let's not forget about our non-coding RNAs, the unsung heroes of the genetic world. Not all RNA has a singular mission of protein coding. Some prefer to operate as regulatory ninjas. MicroRNAs are like skilled assassins, binding to messenger RNAs and keeping protein translations in check. On the grander scale, long non-coding RNAs take on various roles from scaffolding to guiding proteins. They're constantly adding layers of regulation, giving our cells the nimbleness to respond swiftly to signals and adjusting protein levels without disrupting transcription. These non-coding wonders play a crucial role in development, differentiation, and even disease. When microRNA regulation goes off the rails, it can lead to a host of issues like cancer, cardiac problems, and neurological disorders. The revelation about non-coding RNAs has turned our understanding of the genome on its head. It's not just a simple story of genetic coding. It's a complex narrative packed with surprises and subtleties that scientists are still diving into. Have you ever thought that all that so-called junk in our DNA might actually be doing something important? Surprise! It turns out that a lot of what we dismissed as genetic debris is, in fact, transcribed into regulatory RNAs. Our genome resembles an intricate regulatory network more than just a boring old list of genes, with RNA strutting around as a vital player in the show. Gone are the days of the DNA monologue. It's all about the ensemble cast now. Now, let's talk about chromatin, because why let DNA float around like a lost balloon? Instead, it's snugly wrapped around proteins called histones like a cozy burrito, forming nucleosomes. These nucleosomes aren't just hanging out. They coil into increasingly funky formations to create chromatin. The tighter the DNA packs itself away into heterochromatin, the less access transcription factors have to it, resulting in a classic case of you can't touch this QMC hammer. Loosely packed chromatin, however, is the life of the gene party. It's basically the ultimate invitation for transcription factors and RNA polymerase to come on over and get the transcription going. As the stars of gene expression, cells cleverly regulate chromatin structure through histone modifications. Think of it as a fancy dance where adding acetyl groups loosens things up for a hop and a skip, while methyl groups can either join the fun or slam the brakes. The resulting mix creates an epigenetic code that essentially whispers secrets about which genes are feeling active or are taking a day off. Chromatin isn't static, you see. It's more dynamic than your friend who can't stick to one workout routine. The structure shifts around during development, reacts to all sorts of signals, and changes based on the cell type. Understanding this shifting landscape of chromatin keeps scientists on their toes. It's essential for maintaining stable identities in cells while also allowing them to adapt to new challenges. But wait, there's more. The plot thickens with the concept of genetic mosaicism. Imagine your cells having a family reunion where not everyone shares the same DNA. That's what happens. Mutations can sneak in during development, leading to patches of genetically different tissue. Together, these patches are like a mixed bag of jelly beans. Sometimes harmless, sometimes a bit wacky, but occasionally causing a freeze-up when they create serious genetic diseases. Cancer takes the cake as the wild child of mosaicism, accumulating a bonkers number of mutations and playing a risky game of genetic bingo. Now, just when you thought it couldn't get any crazier, we enter the world of chimeras. These creatures, filled with cells from multiple genetic sources, might be popping up around your neighborhood without you even knowing it. Sometimes twin embryos fuse together, creating a single entity with dual genetic identities. And believe it or not, organ transplant recipients are considered chimeras too, carrying donor cells around like souvenir t-shirts. From the womb to the lab, chimeras are a blast. Research scientists are busy injecting cells from one species into embryos of another, leading to animals that could ultimately help in human organ transplants. While the ethics of creating these playful mixes are still up for debate, chimerism completely redefines what it means to have a singular genome. It's all about embracing complexity, layering our understanding of genetics, identity, and what makes us, well, us. In vitro fertilization is like a VIP pass for embryos, letting them strut their stuff before they even hit the stage. Pre-implantation genetic testing dives into the cells of these tiny potential humans, hunting for chromosomal glitches or pesky genetic mutations. For couples nervously twiddling their thumbs wondering if they'll pass on a genetic disorder, this high-tech wizardry offers a glimmer of hope, allowing them to pick the healthiest options for implantation. It's a remarkable way to dodge inherited illnesses like cystic fibrosis or sickle cell disease. Now, let's talk about some serious ethical shenanigans this technology conjures up. Sure, it's great that we can sift through embryos trying to find the healthiest ones, but it raises some heavy questions. What's the criteria? Who decides what's a serious condition worth testing for? And should parents get to play God by selecting traits like smarts or good looks? This slippery slope has some folks seeing red, worrying about the rise of so-called designer babies prancing about with their perfectly curated genes. Regulations about this embryo evaluation circus do a little dance around the globe. While some countries roll out the welcome mat for pre-implantation testing like it's the hottest new trend, others clutch their pearls and impose tight restrictions. The tech itself is as neutral as a Switzerland postcard, but how we choose to apply it shines a light on our values as a society. So the debate rages on, as reproductive choice goes toe-to-toe with sensible ethical boundaries. Let's not forget about mitochondrial replacement therapy. Talk about a family affair. This technique aims to tackle genetic diseases caused by the mother's mitochondrial DNA, which is a hefty burden since she unknowingly passes those mutations to her offspring. In a twist worthy of a soap opera, 3 contributors get involved: the mother, a donor, and the father. The mother's nucleus is transferred into an egg from a donor, sans nucleus, but packed with healthy mitochondria. Fertilized, it creates a child who's a cute little mishmash of genetic contributions. These 3 parent babies have sparked a heated debate, mainly because people are wary of playing with human DNA like it's putty. Questions about safety, who the real parent is, and what it means to tinker with human genetics all bubble up beneath the surface. While some countries have stepped cautiously into approval, others have flat-out banned it, reflecting society's reaction to the very real dilemmas that accompany advances in genetic technology. Genetic diseases, however, are the unsung heroes in all of this chaos, providing valuable insights into the very essence of normal gene function. When something goes haywire and causes a disease, studying the disorder shines a light on what that gene normally does. For example, the puzzle pieces of sickle cell disease unlocked the critical structure of hemoglobin for red blood cells. These rare genetic conditions serve as spontaneous experiments that not only help us understand biology better, but also lay the groundwork for drug discoveries benefiting the wider population. Seeing the impact of these rare conditions brings us right to the forefront of technology development. 2. The pursuit of diagnoses and treatments has fueled progress in sequencing, gene therapy, and personalized medicine. Once considered a sci-fi dream, gene therapy is now making waves with actual success stories for inherited blindness, spinal muscular atrophy, and various blood disorders. It's thrilling to see these advances sprout wings like Lux Turner swooping in to treat a form of inherited blindness with a working copy of a gene delivered via a friendly virus. Hold onto your hats. The future of genetic medicine is popping off. Patients are experiencing life-changing improvements in their vision, with some finally enjoying the freedom of navigating independently. Enter Zenma, the superhero treatment for spinal muscular atrophy, a nasty little condition where motor neurons decide to call it quits. The genius of this treatment lies in delivering a functional copy of the SMN1 gene. Thankfully, when infants receive it early enough, they can develop normally a stark turnaround from what used to be a rather grim diagnosis. While these advances feel like a miracle, there are hurdles to overcome. Gene therapies, while groundbreaking, come with a hefty price tag that can easily skyrocket into the millions per patient. Yikes! And let's not forget that these therapies aren't one size fits all. Some patients don't respond, and side effects can pop up like uninvited guests. Nevertheless, they are shining proof that genetic diseases can indeed be treated, a notion that was like science fiction just a generation ago. Now, let's not skip the CRISPR bandwagon. This spunky gene editing tool is skipping out of the lab and strutting its stuff in clinical trials. From sickle cell anemia to certain cancers, CRISPR is being tested to zap away pesky mutations. In ongoing trials for sickle cell and beta thalassemia, patients have their blood stem cells removed, given the CRISPR treatment, and then sent back in with some upgraded genes. The early results? Think hemoglobin levels through the roof and no more transfusion score. What's even cooler is how CRISPR is turning the tables in cancer research by modifying immune cells to fight off tumors like an Avengers team on a mission. There are also trials targeting inherited blindness, where doctors deliver CRISPR straight into the eye in hopes of correcting mutations in retinal cells. Caution is key, of course, as researchers are keeping a close watch for any off-target effects. After all, no one wants CRISPR hijacking the wrong DNA party. Shifting gears, regenerative medicine is another hotbed of innovation, all aimed at repairing or even replacing damaged tissues and organs. Genetic reprogramming plays a starring role here, helping scientists manipulate gene expression to coax cells into changing identities. Imagine turning adult cells into those magical induced pluripotent stem cells. These cells are like faithful minions, all set to become neurons or heart cells when called upon for treatment. Even more astonishing is direct reprogramming, which converts one type of cell directly into another without a detour through the stem cell stage. For instance, scientists are transforming fibroblasts into neurons and cardiomyocytes, all with a sprinkle of specific transcription factors. It's like a cellular makeover show. But like everything fabulous, there are challenges to tackle, including ensuring these reprogrammed cells play nice and don't turn rogue. And let's not forget about organoids, those tiny, organ-like marvels grown from stem cells in labs. These cuties can self-organize into miniature brains, livers, or kidneys, making them perfect models for research and drug testing. Brain organoids, for example, can develop neurons, making them ideal for studying conditions like Alzheimer's and autism. They provide valuable insights that researchers wouldn't easily get from traditional methods. Not only are organoids great for scientific quests, but they're also ethical heroes that reduce the reliance on animal testing. Plus, since they're made from human tissues, they offer a more accurate reflection of human biology. And guess what? They can be personalized too. Talk about a biomedical dream come true. With all this progress, the future of medicine looks bright. And who knows what else is on the horizon? Organoids, those tiny clusters of cells, are doing some impressive things in the lab by mimicking human organs. Scientists can grow organoids from a patient's own cells, which means they get to model diseases and screen drugs tailored specifically to each person. While these mini organs aren't ready to replace the real deal after all, they don't have blood vessels or immune cells. They are perfectly positioned to bridge the chasm between boring old cell cultures and complex living organisms. Talk about a game changer for research and therapy. As genetic technologies evolve faster than a cat meme goes viral, ethical dilemmas are taking center stage. We're not just talking about privacy and consent. These are serious issues that require a significant amount of head scratching. The big question emerges. Just because we can edit genes, does that mean we should? Genetic data can reveal intimate details about health and even spill the secrets of your family members. So, balancing the exciting benefits of genetic research with privacy considerations is not just tricky. It's a tightrope walk. Disparity is another pesky concern that refuses to budge. The lavish price tags attached to some genetic therapies pose an inequity challenge. Let's face it, genetic tech should be as accessible as that overpriced coffee everyone loves, not just available to folks with deep pockets. Ensuring that everyone benefits from genetic advancements, even your neighbor who awkwardly pets your dog, reflects our society's fundamental values of fairness and justice, making the world a better place for all. Now, let's dig into some mind-bending territory. Gene editing, especially germline editing, can potentially alter the DNA of future generations. Yikes! This notion brings a storm of questions. Who gets to decide the acceptable edits? What happens if someone goes rogue? To sort this out, it's crucial to have scientists, ethicists, policymakers, and the public chit-chatting around the table, hand in hand like a cooperative choir. Only by involving everyone in these conversations can we hope to navigate these challenges with responsibility and thoughtfulness. Public engagement might sound stuffy, but it's anything but boring. Genetics impacts everybody, yes, even your grandma making her famous casserole. By fostering open discussions and making complex genetic lingo accessible, we can demystify the subject. When people get educated about genetics, they're empowered to make informed decisions, steering clear of misinformation and fear-driven stigma. Clear communication isn't just a perk. It's the backbone of trust and meaningful interactions. Let's not forget that genetics is a shared playground. The choices we make today are seeds for the future of medicine, agriculture, and society at large. Bringing diverse voices and experiences into the discussion not only enriches it, but also helps ensure that all perspectives are taken into account. The future of genetics demands insight and compassion, with humanity's best interests close to the heart, guiding us towards a brighter tomorrow. Lastly, the journey of genetics has been a wild ride, with each discovery, from Mendel's peas to CRISPR, paving the way to incredible advancements. The field is growing faster than we can keep track. Personalized medicine customized for unique genetic makeups is knocking at our door, and we can dive into ancestry roots and genetic research like never before. Sure, challenges remain, and ethical questions loom large, but the trajectory is undeniably bright. Genetics will deepen our understanding of life itself and open doors to healthier lives and thriving communities. As the plot thickens, we find ourselves weaving new chapters filled with curiosity about the world of genetics, a delightful realm that happily plugs us into the universal connector of all living beings. Picture this: humans sporting similar genes to flies, yeast, and even bacteria. It's like being part of a colossal family reunion scattered across billions of years. Who knew our great-great-great-grandparents included a humble worm? This interconnectedness teaches us anyone's genetic quirks could spill family secrets about human biology. Deeper into our genetic saga, a splendid revelation bubbles to the surface. We're temporary caretakers of an ancient message. Each gene we carry is akin to holding a baton in a very long relay race, sprinting back to the dawn of life. We're custodians of knowledge etched in our DNA, and how we handle this genetic treasure affects not only our buffet of choices but also the future generations. This genetic tapestry whispers of responsibility, what will be our legacy in this colorful web of life. The cosmos of our body. Each of our cells houses a multi-part tale. Starting from the epic moment two gametes collide pop, the party begins. Since then, those remarkable genes have been dividing, multiplying, and dancing through every cell, carrying a cherished memoir of our lineage. From ancient migrations to encounters with Neanderthals, our genomes are like history books written in molecules, chronicling every twist and turn of our ancestry. But here's the kicker. Our genetic story isn't just a historical or whimsical affair. It's also our gateway to the future. Our DNA does more than reference where we've been. It also nudges our health, our traits, and how we handle life's little surprises. Understanding our genetic blueprint gives us a map for navigating health choices and family plans, allowing us to be proactive stewards of our genetic destiny. Now, let's sprinkle some linguistic flair into this genetic journey. The genetic code acts like the ultimate instruction manual, where DNA sequences become our life's guidebook. Think of genes prancing around like clever sentences, while genomes hold encyclopedic wonders. We've been granted the delightful task of learning this intricate language, and what a learning curve it's been. From the clutters of organic chemistry to the elegant dance of molecular physics, this code fuels the beautiful chaos of life. In a plot twist fitting for any modern tale, our ability to read this genetic language has brought us the power to compose it too. Gone are the days of merely surveying the landscape. Now we can edit, tweak, and concoct novel organisms. However, wielding such power requires a hefty dose of responsibility. Just like any trusty tool, genetics can tilt towards creation or chaos, defining choices in profound ways. As we continue this whirlwind adventure through the intricacies of genetics, let's move forward with wisdom and humility, paying homage to the complex code threading through every living organism. It's a breathtaking dance of life, fraught with possibilities and responsibilities that are both exhilarating and daunting. So, pause and marvel at the exquisite code within, for it encapsulates the wonder and fragility of existence itself. As we wrap up our genetic adventure, It's clear that we've journeyed through the fascinating realms of DNA, CRISPR, and Mendel's legendary pea plants. These venues, from the double helix's spiral embrace to the watering can of genetic tools, have taught us that genes are not just tiny bits of code. They are the architects building the castles of who we are, connecting us to the tapestry of life that envelopes us all. Take a moment to appreciate that the genome lurking within you is like a fine wine, perfectly aged through eons of evolution. Imagine it as a cocktail mixed with flavors from every generation, an extraordinary blend that has never graced the earth before, nor will it again. Each of us carries a flair of uniqueness, coupling our existence with the whole universe, like a funky dance move that no one can replicate but everyone can appreciate. Now picture yourself lulling into tranquility. Your eyelids become featherweight, and your thoughts float like clouds in a gentle breeze. Close your eyes and invite the soothing calm of sleep. Inside every tiny muscle of your being are trillions of cells, all whispering the same genetic masterpiece, a script written in the exquisite language of life. Each DNA strand, coiling like tiny serpents, is the hidden blueprint ready to unravel the mysteries of you as sleep serenades your senses. Let's pause and embrace the poetic lineage of your genes. They are the worldly travelers, cherished possessions from parents to grandparents, showcasing a legacy of survival through thick and thin ice ages, droughts, epic migrations, you name it. Imagine the stories they carry, each connection creating a thriving link in the great chain of life. As you drift further into sleep, recognize that you are a shimmering thread woven into this intricate fabric of existence. The narrative of your genes doesn't just rest in biological archives. It lives and breathes anew within every cellular repair, promising potential to future generations. Life is a dynamic story that never ceases, evolving and adapting like an energetic dance that ebbs and flows. This is genetic resilience, showcased in molecules that have been spinning their tails since life first flickered into being. Now, as your thoughts become soft whispers, embrace the knowledge that you're part of something timeless and majestic. It's nature's old trick to remind you of your connection to the universe, beautiful, complex, and just a tad mysterious. Allow yourself to float in that understanding, as if wrapped in a warm, cozy blanket of curiosity. If you find yourself clinging to wakefulness, feel free to stroll back through any delightful chapter that brought you joy or peace. But if you're surrendering to the tender